everyone.
Yeah. So this is Geodia field, Geodia henscheli and Geodia parva. So we have a bunch of Geodias. We have some particulate feedings like anemones, some carnivorous sponges. So plenty of food in this area probably.
And we are right now at 76 degrees north at an unnamed seamount. So we're exploring something that has never been viewed by anyone before, and we just found this really nice steep cliff with a lot of anemones on it. Also in between you see a lot of worm tubes, and in between these potato-like sponges. Those are all the geodias that Julio was mentioning. Yeah.
Potato-like. I like that. Some asconemas to the right. Those are glass sponges. They barely have any tissue. It's only glass skeleton made of spicules, super huge spicules.
And even more anemones coming up. Wow.
Yeah. Geodias, potatoes and anemones all over the place.
They seem to like fit well together.
Yeah.
And in the corner we see carnivorous sponges, the tree with a lot of branches.
Potentially, yes.
Potentially.
Yeah, it looks like a Bestopluma to me. So carnivorous sponges are very diverse. Ideally, to identify these species, we would need to collect a little bit and to check the spicules, which are the skeletal elements, microscopic, that tell us which species of sponge we have in our hands. But this looks pretty much like a Bestopluma. I'm not 100% sure about it, but yeah.
Even more anemones coming up.
Here you can see how important it is to compete for space in the sea. As soon as you have a hard substrate and food on the area, the substrate, the ground is completely covered by organisms, and they compete. They grow on top of each other to see which one gets more food.
And the depth we're currently in is 1,630 meters, but yeah, we're climbing up as we speak. Sea stock? Oh, what were you saying? Oh, there's a chunky one. Yeah.
Then you have this Solenogaster complicata, this
our spaghetti sponge.
This little bush, the white one.
Yeah.
Super common in this area, the banana hole. We've seen it almost in all the dives and in all the bathymetric ranges.
And actually we've left the banana hole at the moment, and so the international waters that we were in before, we're now much closer to Svalbard than we were before. So we went quite a bit more north.
It's more chilly here.
Yeah. It is. Maybe 4 degrees outside or so, I think, this morning.
We spot whales? No, not whales.
Yeah, whales last night. Yeah, when I was asleep, at least. We had northern bottlenose whales really close by, a pod of them. They are really deep diving whales. They can go to almost 3,000 meters of depth, so almost twice as much as we are at the moment. Yeah, we've also seen, I think, on our last dive, squid around, so that's what they're hunting. Yeah, so it seems to be a popular area for this. So, what more?
That seems to me like a different species.
But it, like the stem looked similar.
Yeah, maybe just one that is not that happy.
Yeah, sad carnivorous sponge.
1,627 meters depth. What is that?
The orange ones.
What's that?
Little anemones? Yeah.
growing on a geodia.
On carnivorous sponges also, no?
Yeah, those geodias are like ecosystems.
Ah, and on yeah.
Yeah,
Now
yeah,
I
so
see the geodia. It was a bit
the potato,
camouflaged. Yeah. There's growing a lot of stuff on top of each other. Yeah, as you said, they're all competing. Looks a bit wild. What are the little white? Also, are little anemones.
Those are anemones as well.
And in between, no, I thought there was a big
There's
worm.
a little Asconema, a little Solenogaster. Yeah, that looks like a worm to me.
Yeah.
We should ask Sergi, the worm guy on board.
Sergi is a worm guy.
Yeah,
I didn't know it.
He used to be. He moved to sponges, but he began working with worms and elites. And I think that he's still doing something from time to time.
Interesting, I didn't know. So yeah, next to Julio we have also Sergi and Paco on the sponge team. And we're at the moment in doing one 12-hour shift, Julio, me, Sergi and Anna Helena. So probably you will also, if you stay tuned, see Anna Helena and Sergi.
We're going for the starboard. Just a little bit. Yeah.
Oh, more anemones coming up.
Perfect. Thank you.
And shrimp. We've also seen a lot of shrimp here in this area. So seems to be a good source of food.
That's a glass sponge to the left, an Asconema foliata, and on top another glass sponge.
The tube.
Yeah, well, I'm not sure. It's really nice.
Mm-hmm. Here at the bottom are Acidia, no? Or is it a sponge?
You want me to zoom in?
Yep.
That looks like a sponge to me. But who knows? Without collecting, sometimes it's hard to say.
Mm-hmm.
Yeah, it looks like a little Hexactinelli to me.
So there's a Geodia with carnivorous sponges and an Hexactinelli there.
Good job, could you step the vessel 20
Potentially.
meters due east at point 1, please?
So the carnivorous sponge seems like a Lycopodina, the little stems that are. I will point them with the laser right now. Yeah, those ones.
Lycoponida?
Lycopodina.
Lycopolina.
There's another carnivorous sponge here. Now we'll be in between the lasers.
Mm-hmm. Very distinct branches.
Yeah, so they are very diverse. And I'm not a specialist on that group, so I may be making some mistakes, but yeah, it's
And a
interesting
sea
group.
star.
Yeah.
And they capture their prey with little hooks and sticky bubbles.
Mucus and specialized spicules that they project to the surface, and then they digest the little critter that gets trapped on that mucus, on that trap. Yeah.
Oh, what are the little balls that are coming up? All these.
Those may be Tentorium semisuberites. Some of them, at least.
Of course.
Some of them.
Tentorium.
Tentorium with T.
Yeah.
Semisuberites. Some of them, not all of them. As for instance, the one to the left. I think it's different. You can see that it has.
Pores. Or big pores.
Yeah, like the body is polarized. It has like a flat area on the top with the holes and then a ringe of spicules on the side. So that I've been seeing it quite often. I don't know what it is, but this one, the ball,
Yeah.
That's Tentorium semisuberites, I believe. Again, to be sure, with the sponges you need to collect and check and check under the microscope and all that, but yeah, it looks like Tentorium semisuberites to me. Okay, and there are some Carnivorous there, so this is a very rich community. Anemones. Shrimp. Many shrimps here. We saw some Roselidae before in this dive, but just a couple. But yeah, here is Geodia, anemone, and carnivorous sponges mostly. Not that many encrusted sponges. Now we just saw one over there, which I will zoom in because I am interested in those kind of cryptic species. Here is where you can find new species. Those little ones that grow encrusted in the rock, they are very hard to collect, more hard to study. So that's why not that many people has studied them. We are not collecting this one here because it may be a little bit difficult.
Terrain, yeah.
A little bit difficult, yeah.
But we see a lot of little amphipods also around in the shrimp, I think.
Yes.
Yeah.
An Assyrian growing next to it.
Tunica.
So I think that the little ones that I told you before with the holes on the flat top, I think that those are polymastias as well.
Oh.
Yeah.
Okay, I will lock that in because I'm here at my logging system.
Or polymastids, yeah, maybe polymastidae as a family.
Yeah.
To be a little bit more conservative. So now the Roselidae, the one I was talking before.
With the sea star on it.
That's the one that dominates in the shoals and in the void.
Yeah, it's getting munched upon.
Here we have two sea stars, not one but two.
And above we have a lot.
Three, yeah. So 1, 2, 3.
Yeah. The one on the very right looks already very swollen from eating maybe on the sponge next to it. On the crust looks a bit holy.
Let's see if I can zoom in without moving this too much.
Yeah, looks like the sea star is having a sponge eating party here. Also the one on the Geodia is like probably munching. So the sea stars have their mouth in the middle of their underside, and there they have like a beak to munch. Yeah, do you think the one on the right has eaten all of that?
Maybe.
Like, yeah,
maybe.
all of the crust.
So echinoderms are spongivores.
Yeah, so the sea stars are echinoderms. Yeah.
Rough copy, thank you.
Okay. Shall
Yeah.
we move on?
Yeah, let's move on.
Thank you. Check it again.
further up we go on this unnamed seamount. So our dive will take us up this cliff, and then we will reach a plateau, but
Got the one.
yeah, depending on if our system updates where we are at the moment, it's still some time, but actually it looks like as if we've reached the top. Yeah, I think that has not been updated.
That's
Yeah.
not updated, right?
I see a little lonely soft coral on top.
Hola amigos.
Hola.
¿qué tal?
No. It goes on, and are those all our carnivorous sponges?
Yes.
The what was their name? I forgot. Julio had asbesto
Avestapluma.
Because I'm not a sponge expert. I'm learning from the sponge experts here. A lot of names and also other characteristica of all the different sponges there are. So that's been very interesting already.
You will become an sponge expert very soon. After 1 month here.
Yeah.
So those are more Rosellide. Yeah. We don't want to say which species because we are not sure, and we believe that this can be more complicated than what we thought before. So before we used to believe that there were 3 species. Now we've been collecting some, and we are suspecting that there are more than 3. So we stick to the family name. That fish is the same that we saw the other day.
They're more whitish, yeah.
It's a bit more whitish.
But we've seen more of those already.
Yeah, I don't think I would catch him.
On camera.
No, it's okay.
Yep.
It's okay, Antonio. Don't worry. Yeah, keep going.
I think the rock underneath also has interesting colorations or characteristics here, so we often see more yellow, orange, reddish crusts, so probably some mineral metallic crusts here on the rock. Oh, and a quick shrimp flying through. Yeah, and a lot of biodiversity on top of it. Yeah, it's interesting because we've been in similar depth further south, but with different communities.
Yes. This is Teloloris favellata, I believe.
The fan-shaped one,
The
right?
fan shape, yeah.
If you could move off, please. Point 1, continuous, please. Due east.
Sorry, fancy.
All good.
Another. So we start the stream. We were at 1,630, something like that. So
yeah.
we climbed up about 30 meters so far.
And how much further are we going? Checking our folder with all the dive profiles and details. Yeah, we will end at about 1,500.
Okay, so still 100 meters up. That's big Rosellide to
Yeah.
the left. Another one here.
Can we maybe go a little more to the right? Yeah. Looked as if the current was more favorable for the communities there. Thanks.
See some good rock on this one.
This is rock.
Some
No, there is one
obvious one.
here on the left. Oh,
There's a shrimp flying in our spotlight. You can see all the little feet dangling.
Rosellide.
Yeah,
Those are
because a leader with a lot of tips.
probably very old sponges. They grow slowly during years and years. So
And they can grow several, like age for several hundreds of years or maybe even thousands.
I'm not sure about these species, but
Sponges in general.
in
yeah.
general, yes, and those are really big. So
And it has a carnivorous next to it.
yeah, I would say that they are old. That for sure, many years.
And next to the living white Rosalida is a dead Asconema or
The brownish,
maybe
yeah.
not dead yet, but pretty
Yeah.
bad.
Yeah, no, that's a dead one. That's Carnivorous.
Providing also biomass.
Well, they help creating a system on the sea ground with the spiks.
Yeah.
They remain,
Mm-hmm.
and then you have this spik mat, which is also an habitat.
Made of
Yeah,
glass.
it's
in
like
the
the
end.
soil in
Yeah.
a forest with the leaves. And
Yeah, nice comparison. Is that a bush of Asbestopluma?
yes,
just a question to
tell me.
you were saying for us to follow the right, but in this direction is a completely different from the transept.
So
The idea is to follow the transit is more to the left. If it's just forget the transit and look below. It's coming.
Oh!
aha.
Oh
okay,
wow! There's a beautiful squid. Can we take the lasers off? Thank you.
Lasers
wow.
off.
Well, now just follow.
Oh, beautiful. Yeah. Beautiful colors.
I understood.
So our lasers were 10 centimeters apart, so this should be
It's a big one.
over a half a meter at least long. Going up. Bye bye. Safe travels. I'll have a look.
So just follow it parallel to the line, and then when you reach the end of the horizontal, let's go up.
So parallel is
Maybe
more to
that
the
was a
left.
gonatos. Yeah.
Let's do that. It's going parallel all the way.
Okay.
Looked like a
See,
gonatos.
there's
Huh?
this-
Oh,
this
that's
rock, if you see a rocky outcrop, try to follow it a little bit, but don't overcomplicate it. It's just, yeah,
and eel pout, is it?
I
No.
think that this is all rock.
Some fish flying through. and good to take the lasers on again if you have the chance. What's lying there? Fish?
Fish.
So we're flying a bit along the cliff and we see a lot more geodia everywhere, even though they're better camouflaged than all the white sponges and other organisms that we see. Yeah, and so many shrimp.
And it's a beautiful landscape.
Yeah, we had no idea what we would find here because this has never been explored before. But yeah, this is really nice. So the rock is forming a hard substrate for many sponges, but also other organisms like the anemones and soft coral to grow upon. And they in turn then provide more substrate for other organisms to grow upon or to feed upon, and so they all attract each other and build a whole habitat and ecosystem.
Going up 1,580 meters, 50 meters more.
Both copies, thank you, Nancy.
Here are more of dead
There are
Asconivorous.
many dead ones.
Circle of life.
I wonder why.
Maybe it was
there's
just the time
Yeah, maybe. Maybe there's a lot of sedimentation here.
that blocks them, or
Yeah.
but the others seem to be doing well, or well enough.
The other
Sponges in general. Yeah.
Well, there are geolias, but not that many other than that. Big species. So you have the geolias, and then you had some Rosalia from time to time. Some.
And those little calcareous sponges?
Those are anemones.
No, that one's down below, you mean?
No, these ones.
These dots.
Yeah. Let me see.
Baby sponges?
They don't look to me like sponges.
But these would be. Oh, hello, fish! You want to be in the camera. Is it a snail fish, maybe?
So, one second, Antonio. We're going now. We
Want to look at the little.
solve this mystery of this kind of pinky to me.
I think it's maybe because they're so white. I don't know. Yeah, hard to focus here or to hold it steady when we are so zoomed in.
Yeah, I don't know. Fancy.
I'll just log them as unknown sponges. We've already encountered a lot of probably unknown, undescribed sponges, and we have quite identified some of them in the lab. What's the latest status? Like how many potentially undescribed species?
3 to 4. 3 to 4. Yes.
Yeah.
And they're quite clear, but yeah, we need to go to the lab and get DNA sequences, and
It will take some more
yeah,
time
it will take some a
until
while,
we're
but
very, very safe.
it looks nice. It looks, yeah.
Keep going.
Yeah, keep going, Antonio, please.
Yeah,
thank you
thank
so
you.
much. So the mystery is not solved. So if somebody has an idea, what are those little...
Write it in the comments. It seems that the carnivorous sponges, if it's indeed this Asbestopluma, is much more popular here than on other cliffs that we've seen. I wonder why they like it here so much.
So I guess it depends on the balance between what type of food is in the water. Now I'm guessing, but carnivorous sponges, they eat living things. These are little amphipods, little crustaceans in general, maybe millimeters in size. So this could mean that this area is rich on this kind of animals. When we see more sponges that are just filter feedings and not so many anemones and not so many corals, that usually means that it's more oligotrophic, meaning that there are less nutrients, and that the food is mostly dissolved organic matter. And then sponges, some species of sponges can still eat that, but not sponges like carnivorous sponges, which eat larger things, and corals as well. Corals eat larger particles or larger food.
Does that also fit together with the fact that we see a lot more shrimp here?
Maybe.
And yeah, some fish at least as well.
Maybe. Yeah.
That like to feed on those.
We are guessing a lot, but yeah.
Yeah.
that's,
Yeah.
uh,
Just hypothesizing here.
those fishes are cool. I like
Yeah.
them.
Yeah. I'm going more lateral left now. Because again we see this.
So
You are going north.
I'm going to.
Not exactly. I'm going almost south because we are facing this edge.
We're discussing with the rough pilots who are doing the great job of steering the whole robot and navigating us through this. What's the best way to continue the ascent along the cliff?
Point 2 continuous.
And depending on how we move with the ROV, it also has to be in line with how the vessel moves. We have a long cable, a long umbilical that attaches the ROV to the ship, and we always need to make sure that it has it's the right amount of slack and that the vessel moves so that we don't somehow make a cable salad. Yeah.
So Antonio, I guess that this kind of landscape is not good to stop. It's not that I want to stop now. In case I see something. Yeah,
Asking for a friend.
we still have plenty of space in our bioboxes, so
Yeah, if you see something interesting,
just
tell me, and then we see if there is any
Are you thinking?
place around to stop.
yes.
Are you thinking about Lysidendorix or about other? What did you see? What's
I'm
interesting?
thinking about this Solenogaster, and maybe if we see some nice loose rock with grass, it would be good to collect that. And
Yeah.
also I'm thinking maybe something for solving.
Our underwater photographer here, who's
Yeah.
doing a lot of great blackwater photography of also samples that we take here. He's creating beautiful images of of different organisms, giving them a real personality when you look at the pictures.
So for instance, Antonia, you see those?
Building also, he built a great setup to make all these photos happen.
You can do it?
You can check his photos out on, I think, on our webpage as well, and or on Solvent's webpage. Solvent Zanke is his name. He's also a marine biologist.
This one is also nice. Oh yeah. Have to give me the option.
And you're right now looking at Lysidendorix for?
And the crust.
And
There's
the
our
crust.
crust here that keep seeing it, and I'm a little bit annoying not to know what it is.
Which one?
That's not acceptable. This one.
The white one, yeah.
So if maybe we can slurp the Solenogaster, we can also slurp the crust. Maybe we can try.
So our ROV has a different setup of sampling techniques. Among others, this slurp, which is attached to the left manipulator, so the left arm, and
With the slurp, you mean?
it can suck
Otherwise the crust.
organisms into different chambers that we have in the back. And we are interested in sampling different Lysidendorix, so different sponges, Lysidendorix complicata, because the sponge team is doing connectivity studies on them. So we're looking at how they're connected from one side to the next, how their genetics look like, because they're super successful species down here from quite shallow depth, like 600 something meters up to more than 2,000 meters. And that's not so common that a species is distributed throughout this high variety of depths. It must be doing something, or it might something that is making it very adapted to all of these different conditions.
Okay, not very stable, but it's fine.
Okay.
So we're taking more samples of these Lysidendorix throughout our different dives.
So that will be
In comparison to other samples where we maybe just take one because we want to know and find out what it is in the first place.
Yeah, just let me have a close up very quickly. So that will be one. Here you can see the little Carnivorous sponge to the right, going on top of a geodia.
And ascidians.
And ascidians.
The transparent organisms.
Would your ROV bring the vessel to a stop, please?
And then I want to see
And there's a little
If you can
amphipod
follow that.
crawling up the sediment.
Copy, thank you.
this one.
Yeah, between the lasers.
Maybe it will be hard.
It's crawling towards the crust. We have also another team of researchers, and they're, among others, very interested in these amphipods and these little crustaceans. We have two big amphipod experts Anna Helena and Anna Nina. Anna Helena will actually probably come soon to take over the next shift. But their team is also looking at a lot of like on command. Anna Helena comes in.
Which one are you going for?
They're also looking at all the other critters that we have down here. Hello, I was just speaking of you. Because we've seen amphipods.
Yeah, no, this is different. So this one, and then this one, or this one or the other.
Yeah, go for that one.
Yeah, I think this
Okay, yeah.
How are you doing?
These walls.
There are
It's
lots
super
of
beautiful, but
arthropods
it's hard to
in some
stop.
of the samples
So
from
they
the water,
tried before.
the previous time.
Nice. You
So-
happy?
Yeah, of course.
Porque arriba
So what we did
This
then
will
was
be
to get
four.
some
okay, yeah.
of the bryozoan mats
Mhm.
that
Like
we
this one.
saw at the end of last time. And they were full of life. I mean, they look
yeah.
kind of brown and dead and everything, but they are really
They're
full of
also
life.
building a habitat there.
And you are sampling Lysodendorix.
Of course.
Are you also
What else?
playing the Spot the Lysodendorix game?
Well, that's like
The
a boom boom. We will.
The what? What are you playing?
Who first sees the Lysodendorix gets an apple.
An apple.
An apple.
Come on.
Oh,
That doesn't.
that's an
Oh,
apple.
You can get an apple.
Chocolate, no?
There is a little bit left in this box.
Oh, I didn't know that was so great. Sweet and weird.
Here, yes.
That's gonna make the.
Oh, I'm getting warm in here.
a second.
Oh, this broke up.
I'll just bring it a little bit back and see. Sorry, this is not. Sorry.
No, that's
Now
fine.
it's second.
Now it's second.
Now it's okay.
Sorry, mate.
It's gone in.
Stop.
So I'm noting down all the information for
Great.
this
Thank you.
sample.
It's in a different menu.
What time is
So
it?
you can control it from here and from there.
Yeah.
Ah.
yeah.
So this is in
What's
chamber
next?
3, right?
Chamber 3, yeah.
This one.
4.
This one, yeah. If
The
it's
same chamber.
same chamber, if it's too difficult, don't lose too much time. Okay?
But this is
Because-
not another
which one?
Lysodendorix. No.
No, don't worry. Hmm?
And more paperwork.
No, lo sabe.
Okay. No.
Lysodendorix.
So this is. Okay. I'll wait a second so that we can make sense of all the paperwork.
I think that if you take a look at the sediment, it will be fine.
Are you second?
Yes.
And now you're doing the crust.
Yeah.
Now for sure. Don't push it too much because we will come back.
Right.
Oh. I have the message for the tape change in 10 minutes. So we're trying now to slurp also the crust, the sponge crust,
Let's see how
of which we don't know yet what it is, and we'd like to investigate it further. Let's see if that
Yeah.
worked. No, it's too
now because
crusty.
it's sideways.
This is a crust.
Yeah,
It's fast.
and it's doing a very good job in being encrusted to the rock.
Yeah, no, that will be too difficult.
Some pieces went.
Some pieces went.
Yeah, I think so. There's nothing there. Still there. something.
Yeah, no, because
Subplan?
It's still seconds.
Oh, maybe we look later if we find a little rock where it's on, and then we can collect the little rock. Because this looks tricky.
Are you trying to bring the entire mountain?
Yeah, let's try. Maybe we can see the metallic rock underneath, metallic crust of the rock.
We should already have. Was not only that.
okay.
Shall we try
Now
to
we have it.
later on?
Now we have it.
Yeah.
Perfect. Now you took a piece.
Nice.
Yeah, now you took a piece. Thank you so much.
Cool. So, can you help me? We had a cast.
One second.
Okay. We're still not done.
Not yet. Okay, stop now. I should be home.
Sorry?
I should be home. I should.
So in this slurp, there is Solenogaster and white crust.
And that was our cast 4. Did we slurp 2 Lizardendrons? No, just 1. Okay.
Just one.
So, and that all went in slurp 3.
Right, yes, exactly. Have a bunch of amphipods. Asconema for them.
Circulates.
Yeah.
Mm-hmm.
And
Yeah.
small stuff.
Yeah.
Was
Yeah, a lot of
took a picture and I posted it.
Cool. Yeah, we also see in between a lot of serpulates, so worms that are building tubes around, hard tubes, Carnivorous tubes, I think. Yeah.
So we found, if you were, yeah, you were there watching it, I think. Small, large, we thought it was sponges, orange ones,
Yeah.
and they are apparently Solenogasters, which are like a group of mollusks. Very special.
Okay.
We
Cool.
have to
Oh.
very
We
preserved.
still have 5 minutes.
Oh,
Yeah, let's
interesting.
go.
Okay.
There's still 5 minutes, guys.
Yeah.
Yeah.
Did you do your stretch on the floor?
Yeah, don't worry.
Is that an anemone that is retracted?
Yes.
When has it?
No, it must have.
Do you want to have fun?
Smell like
Huh?
let me toilet in.
Okay, no, I'm fine.
Yeah, they
Just
say.
if you want.
well.
No, okay.
Do you want me to jump in from swap?
If you want, I'm okay. But if you want to,
If you want time out, so you'll jump in if you want.
so we need umbilical.
You're selling fish.
Is it okay?
Yep.
There's a
Yeah.
It's one of these that has the arm. Yeah. And I was just wondering whether you have any, or you need this arm.
We've also seen really cool solutions going on.
This
Yeah.
arm that has this.
Gonata, maybe.
Like this. Is it big crabs?
And there are lots of shrimp everywhere.
Like
Oh yeah, now we can see them. They're all in the water column, and probably, yeah, they find a lot of food here.
yeah, it will be. The hill will be on our right side, but if you want to follow the line,
Maybe a little bit of blue.
And we're talking a bit about the next directions for continuing
it should be roughly
the ascent here on the summit of, yeah, or at least plateau of the seamount.
It's quite nice because
And then we are more aligned.
Yeah, more or less.
So we're swimming through all the shrimp and little marine snow organic matter that we see here.
Yeah, that's one.
And another beautiful rock wall coming up with many anemones and geodias, the
Yeah.
potato sponges.
Again, the Amphidiscola.
And
The Hexactinelli with the stem, Procelides,
The worms in between.
plenty of geodias. I'm amazed how many geodias are here in this world.
Right, and then this doesn't seem to be quite wide, so it's not working.
So we may answer these kind of questions: why here there are geodias and why geodias are not in other places, and in other places we have plenty of Hexactinellids. This you can try to explain checking the environmental factors that we are gathering all the info from with the CTD, which is a device that is in the ROV, constantly collecting the temperature, the salinity, conductivity,
Oxygen,
oxygen.
Right now it's, for example, minus 0.5 degrees cold here.
Quite cold.
But mostly it has been minus 0.6. So slightly warmer. Even though we're much further north than during the previous dives.
It's a big one over there.
Big carnivores, or you mean the one behind?
The
on
Hexactinellids.
The Hexactinellids,
See?
yeah. On
Looks
such a
like-
tiny little stem, that's amazing. And big carnivorous sponges here, not the
Yeah.
asbestopluma, but different ones, right?
More anemones, so it looks like what we saw at the very beginning.
Yeah.
That beautiful rock with
Mhm.
completely covered with anemones and carnivorous sponges.
They have such long tentacles, these ones.
Yes. Do you have an idea about the species of the anemone?
I can check if I can get an idea. Oh yeah, 1 minute till tape change. We'll change the tape soon, in less than 1 minute, but I'll let you know.
Yeah.
Oh, there are a lot of anemones that look similar here. Maybe Omatidia? They seem to have long tentacles. So when we change the tape, that means that we change the recording of the video, and we always save it after 2 hours to have backups in case anything crashes. We never lose too much data. And oh, can we stop the recording?
Yeah, ready to stop now?
This is nice.
Yes,
Yes.
stop. Yeah.
Okay. Stop.
And after 2 hours, we also always change the shift so that we can get a little break or continue, for example, lab work. And then Anne-Helene and Sergio will take over.
Okay, have to start recording?
Okay, start recording again.
now.
Yeah. Thank you.
This is really nice. It's a good moment
You don't want to leave?
to change. Now we are here nice and steady.
Yeah.
Yeah.
Shall we?
Let's have a nice lunchtime.
Thank you.
I'll finish
Enjoy the
lap,
dive.
actually. So
Just grab all
we
my
can go to the
stuff.
microscope.
Looking at sponges
Yes.
under the microscope. All right. Bye bye.
Practicing it and see any change in the flavor and the positions.
Oh, lots of stuff flying around you.
Because we are slightly cold in here, right?
Yeah, but it's now 18 degrees.
Yeah.
Oh,
Yeah.
wow. That's a lot warmer. Hi all, and this is
What's that the other option?
Sergi and Anna-Helena fresh in from the lab. So directly from the lab where we have been looking at the samples from the previous time, and we are still processing them. It takes a lot of time to get through all of the samples from a dive. Lots of noise in the background here, but we are continuing up the hill.
1,500 meters, right?
Yeah, we are about 1,544
Just tried to
meters.
set it twice on
And
that. Just tried in there.
Hansi said we have minus 0.5 degrees in the water, and that's colder than it was this morning, a little bit further north,
Yeah.
like 20 minutes of steaming further north, not very further, but there we had minus 0.2. A little bit higher up as well, that was, so not super comparable, but this is a beautiful wall, and it's almost a vertical wall.
Yeah.
Loads of anemone,
Yeah, we're still climbing.
and I am happy to click the anemone button.
Well,
And we see also carnivorous sponges here, right?
yeah, some of those carnivorous sponges, these white
Yeah.
branches, like the really thin branches that you see. Sometimes you see them, you see the lasers now, at the very base of the lasers, there's these carnivorous sponges. And there's a few there of them.
That are more branchy, right?
Yeah. Let's have a closer look at this edge, where you can find these carnivorous sponges that you see. These
And
thin
also
branches.
it's Geodia, right?
Yeah, it's
No,
plenty
I
of
think he's creating
Geodia species. Oh, well,
once again,
some Geodia Henshelli, which are these roundish sponges with the oscula, yeah,
One osculum at the top.
in the middle. Yeah, the oscula is it's a opening where the water flows after it's been filtered.
So the out opening.
Yeah,
What are the in openings called?
porous.
Porus.
Yeah,
That's the basic for the porifera.
yeah.
Yeah.
But there's some sponges you see here where the lasers are, which are pretty colonized by other organisms. If you see the lasers,
So that's a Geodia
yeah.
with carnivorous sponge
this is
and an anemone, right?
yeah,
And
other smaller sponges too.
are all of those white tubes? Do we think that they are serpulids or?
Not the ones on the sponge,
But there
the
are some
ones
wormy things
beside,
on.
Yeah, definitely.
So that sponge is a whole little ecosystem in itself, even
Yeah.
before we start to look at the
Yeah.
microbiology. That is also a complete ecosystem, right?
Yeah.
So on very many levels, this sponge is an ecosystem. Okay, those 18 degrees were not that super warm actually.
Yeah. Okay, so
really love all of these anemone.
Uh-huh. They're bringing color to this
Yeah.
wall.
Together with the shrimp, there
Uh-huh.
are these popping pink
There's a
areas.
sea star in here, you see, orange one. We're still climbing. I'm still surprised on how these Geodia organisms they made it. I mean, they really like the vertical walls.
But these are some Rosalida as well,
Yeah,
right?
some versatility.
In between everything else.
I'm going too fast.
I'm so happy I
fine.
found
I
the
think it's
Carnivorous
fine.
sponge button, because now I don't have to type all the time.
Mm-hmm. Yeah, there's a bunch of carnivorous sponges, meaning that there's lots of food for them,
Yep.
meaning crustaceans.
Ooh, you're rubbing it in again. And I just said from the previous samples that we took this morning, I've sorted out quite a lot of amphipods, tiny little ones, and I guess they would be the favorite food of all of these carnivorous sponges.
There were some big ones, right? Too.
Yeah, there were a few big ones, and most of them were in the family Pardaliscidae, for those of you who are really amphipod nerds. I think 1 or 2 different species there, which would be normal for this area. But already in the smaller size fraction I had 5 or 6 different species just in one sample, and we didn't even sample for them. This is just what comes with the sediments we sample, or with the bryozoa mats that we sampled earlier. And I think maybe we're seeing some of these bryozoa mats here together, maybe with spicules from
I
Geodia.
think those are the ones, yeah.
Because I can say, when I looked through those mats that we sampled, that they were a combination of spicules from, I would guess, Geodia, and lots of old bryozoa.
Yeah, we don't see the bryozoa, the living ones, right, in here?
No, so maybe that's just a spicule mat then, because there is no lacking of sponges here.
And just below, we see. We'll see in a minute. Sorry. This group of serpulids. These worms. They are building a structure in here. You will see in a minute.
Yeah, they're almost building their own little area, right?
So the white tubes that you see those are tubes of worms, a group of annelids, which are called serpulidae. And but there's some living, and there's some that are dead. The white ones are the ones with animal inside still. They are alive. The other ones, the brownish organisms, remains of the tubes. Those are the ones that are dead and are producing a substrate where all the ones are being. Oh yeah.
What's the yellow thing on a stick?
Let's find out. I'm not sure about it. I'm not sure we have seen these before. Probably a sponge that got there. I don't think the sponge built this the stalk. Probably colonized the stalk of an organism that died.
That's a good way of doing it. But this is also quite common in nature, that there is no unused habitat. If a habitat becomes available, it will be quickly utilized by somebody.
So you see at the right hand side there's some of the remainings of these tubes of circulates. They're producing a substrate
Yeah.
for others to be colonized. And this sponge, you remember the name of this sponge on the top right?
Oh, that's an Asconema foliatum.
Yeah. Yeah.
I am getting it. Now we have to just find the species of anemone because there's so many of them. I
Yeah,
think Fancy
that's
was
right,
trying to
the
find
one.
out. She had some suggestions and I didn't hear it, so maybe you guys already heard it.
Some shrimps. Yeah.
I think they're actinostolids. But there are many actinostolids, so who knows?
Yeah.
Probably remove 0.5, that was 1, 2. Continues until it catches you.
Ooh, that's...
Can you wait a second?
Well, this one we need to look at. I
Yeah.
would
Good job.
think...
We could put a move in, please, bearing 045 at point 2 continuous, please.
Yeah, we saw something interesting, huh?
Yes, this is
Yeah.
bottom-living jelly. It is... Oh, I remember and I forgot, but I can find the name here.
So I'm gonna let the ROV get stable. Oh, look at that! This orange
Yes.
stuff in here, we know now is not a sponge. We know now it is a Solenogaster, which is a group of mollusks, which is not that common, I would say. It's more common in deep sea areas. And yeah, we're gonna have a look. More detail. Really long. I was just measuring it after taking a picture, and it's like it was a list. The one that we collected more than 4 centimeters, and I think this is one. Yeah, I bet this is. What do you
So
think?
oh yeah, let's collect some of those. And let's also collect the Lucernaria that we saw.
Look, it's moving.
Yeah.
Looks like a snail. It's not a snail. It's a mollusk.
It doesn't, it's not a worm, but it's a mollusk that looks like a worm.
Yeah.
Probably,
Yeah.
The
Shall we collect
Norwegian, yes, please, and the Lucernaria.
The one on top. Yeah,
Yeah.
we'll have a picture. We'll.
Let's first have a proper picture. Can we maybe also without the
Shall we slurp first this and then go
yeah,
with the
but
other
let's zoom in on it without the
Yes, it's just
one? With the
Lasers?
lasers.
5 at point 2
Yes,
continuous
the-
until it catches up. Okay, slow down a bit.
So on the right, there's a worm, kind of a worm. Then we'll go for this one, which is really beautiful.
We first want to just film it a little bit, that thing that looks like a worm. And it is a mollusk, so it is close to snails and bivalves and octopuses and those animals, but this is covered by
Yeah.
little scales, so it looks almost like it's covered by fur
Oh, yes, please.
when you look at it in a microscope. Can we take off the lasers a little bit? I can do the nose.
Yeah. Yeah, that's
is somewhere.
The other one, you look at
Yeah.
the other one. I don't know. Probably there.
It's
Yeah.
under here. How do we want to slurp them, right?
Yeah. I think
Do we
that's-
think that the Lucinaria will survive to be slurped?
I don't know. Probably. You can just give it a try.
It's a jelly thing,
Ah.
so maybe not. But this one definitely will survive to be slurped.
It's a vertical fish.
Oh yeah, you cannot do it.
Go on, but you have to be.
Then we will just look at them. Okay,
Yeah.
can we go and look a little bit up then at the
We
Lucinaria?
have another one.
Yes. I mean, you can try, but it's just because it's vertical, you might
Yeah.
fall away.
Just then. Have a look. Right.
There,
Ooh,
look at that. And if we manage to just have that in the picture a little while. So this is a jellyfish that is attached to the bottom. So this is a benthic jellyfish. It's called Lucinaria. Let's see if we can suggest.
So I'll stay here so that we can film it.
So the suggestion for a name in the area would be Lucinaria bathyphylla. And it's quite cool to see.
So it's like a jellyfish. What is that?
It's a jellyfish, but it is attached with a stem to the bottom.
Uh-huh. Let's have a closer look. Oh, look at that. So it's got short tentacles, and in these tentacles they have these cells that when a prey gets closer and it touches it, it gets activated. The cells are activated and they sting.
So like all other jellyfish,
Yeah.
they have stinging cells, and it's also together with the anemone and the corals that we are seeing both on this dive and everywhere else in the world.
Mm-hmm.
What do you think that long, thin thing below it is? Do you think it's a worm?
Yeah, probably, yeah. Things below of these beautiful specimen.
Yeah, but that's a beautiful thing.
Mm-hmm.
It's like it has these little. This really has some small pom-poms, right?
Yeah.
Going out, back in again. And you don't think it's possible to make a sample of this?
You could, I mean, it's vertical, so you have to free fly it and
Yeah. Okay.
move the arm as well. You can try like
We no. Let's
Is it
let's.
slurp or-
Yeah, we'll be with the slurp.
Yeah, yeah, if it's slurp, I mean, you've got a fighting chance if you slurp it.
The
Yeah.
thing is that, well, the slurp is going to be
I think it's
The
going to
thing
be
is
quite destroyed if we slurp
damage.
it.
Yeah.
If you bring it out and free fly it in, you should be able to get it.
It's fine because they're so soft.
Ah,
And then
Ah.
when you slurp it, then probably it's going to break
Yeah.
into 1,000 pieces.
Would it be a scoop or?
A scoop would be the best, I think.
Yeah.
Hmm?
Still, we can make comment just to see if it's.
So this is really an interesting little place right
Uh-huh.
below the and so on a little ledge between two vertical walls here,
Look
right?
at this beautiful wall.
Yeah, look at that
Just
wall right
waiting
above.
for us.
Yay.
Was it on there?
So we're gonna try to.
Now, for all of you who were rooting for the University of Bergen Manet team this morning, when you were saying we should try to sample Echinoderms and stuff, this would be this is a picture I already sent home to them. So we are thinking about them. Whenever we see things, we think about all the different colleagues we have that would like to work with these animals we are seeing. So this is definitely a collaborative work. Whenever we find something and sample something, we don't only sample for the 6 of us, 8 of us on the vessel, but we sample also for colleagues who are working on specific groups. And that even goes into the nerdy little details of that genus is a specialist genus for that person. And sometimes it's big groups, sometimes it's small groups, but this is how the entire community, I would say, of taxonomists work. There is so much life to work with and catalog and discover that we don't need to fight about it. We all have our specialities. And we still can collaborate very nicely, so.
Yeah.
Stay fresh, jumping arrows and be there. To the leadership tonight. Where do we scoop?
Yeah.
You can't hit the rock and just flick it from underneath.
Yeah.
Let the rock pushes off.
So yeah, we are considering which is the best approach to get the sample. This is not. It's quite challenging in this case because it's almost a vertical wall. Then the ROV is standing, it's flying. We're gonna try to get it using a scope. Because probably with the slurp it would have been easier to collect, but we would definitely damage the specimen. This is something that we don't want to happen.
Yeah, but that's an amazing animal.
It is. Just another we are.
So still, it's
Yeah.
very nice to zoom in on this one, yes.
By mistake. Yeah, he's used to this as well, so I'm not.
If you drop it, it's a long way.
Do you want to do it?
Yeah,
go on.
just
right in the center.
I know,
It's a beautiful
It's sitting
specimen.
there pretty much stable at
Yeah.
12% first one, but I can try, just try and leave it just doing it. If I can really hit the scrape it. Yeah, it is.
So again, our amazing pilots are going to give it a try.
We're going to zoom in the.
Yeah, I'll just get a scoop first.
Yeah.
Okay.
Is that attached to that rock, do you think? Or
Probably. yeah.
Maybe, yeah.
I'll have a quick look first, rock.
Okay, and then we'll have to stop.
Well, wait, I'll have a quick look at this rock first just to see if it's attached.
Ah, yeah.
Rock.
probably not attached. Well, it's probably attached to the rock.
Can you zoom in on your camera shot, please?
I think we're way too much in the
And go to
front.
the
Oh,
right.
yeah.
Oops.
There's like a really small stone there.
Oh.
He didn't like that,
Uh-huh.
but that's okay. We can bring him back.
Oh, shit.
You are sampling like you're picking flowers. Oh, no.
Oh, no, yeah. It's actually stone on it. Try to get the smallest that grabs as well.
Oh, it's flying now. Oh.
Ooh.
Like this. It's fine.
Ooh.
My catch.
Oh no.
Ah,
Oh. Oops. Sorry,
flight. It's at the front there, but
so it managed to escape.
Wow.
Struggle down.
Yeah.
Swamp.
Okay, these creatures. They
Well,
know
it
how to-
deserved to stay.
Oh look, it's flying. It was flying on the left hand side. Ah, yeah.
So, okay.
Don't worry.
Didn't know where to come with.
We got a beautiful film of it, and it will find another rock to attach to.
Mhm.
Okay.
No problem.
Thank you for trying.
Alright.
Now we are at the base of this
Turn the laser on.
beautiful.
Yes, please.
Laser's on.
And let's fly up this very beautiful vertical wall.
I know,
Got
yeah.
it the first time.
Crush this.
Okay, all the species, Serpiolids, Lisodendrorix, the anemone, sea stars. This is a community we saw yesterday, right? In the Schultz Bank. I think so. So they were, look at that. Look at this density of anemone.
Yeah, look at that.
Let's just stay here for a sec. That's indeed really beautiful. There's some Rosellidae also here.
I am going to dare push my anemone button.
Yeah.
I did it. I'm going to do it again.
Deserved.
Another. Just to indicate we have quite a lot of them. And some carnivorous sponges.
Yeah.
And my eyes see Lisodendrorix.
Yeah.
Yes.
It's been a minute
Do I
since
win
we
the
saw.
apple for this round?
Yeah, but what's the name of this beautiful creature? Luternaria Bathyphylla. Yeah. Bathyphylla meaning that it's a really deep organism.
Yeah, it likes
right?
to be in the deep. Bathyphylla. Loves the deep. Oh, but this looks almost like a painting by
Yeah.
Van Gogh or something like that, right? This is really You know, like that Starry Night from Van Gogh? Van Gogh?
Yeah.
Yeah, the Starry Night. It's a
Yeah.
little bit, if
Yeah.
you made all the stars pink, I think it would be there.
Not smash it completely.
Is that the tulip sponge or is it something else?
The one right. Yeah, I think it is.
Tulip sponge?
Yeah. At the
I'm
very
definitely getting the
base.
apple of this. Oh, and a fish.
It's found.
Right, Geography, if you can all start now, please.
It's found a place where to hide.
Yeah. But this is a good place to have a lot of cracks and crevasses and everything, so you can hide in there. And of course, we have tons of Geodia. So are all Geodia kind of smelly when they come on deck, or is it just the shallower? So
I don't
like
know.
Geodia, Macandrevi.
So Macandrevi is predation smell.
Oh yeah. Or you can smell it on the entire boat when you get a load of those up.
Yeah, it means that they have like chemicals, right? And Paco, the other sponge person, so we have Julio, Paco, and myself studying sponges. So Paco is interested in studying the chemicals that sponges produce, which may have, in some cases, some application for new drugs or new methods.
Yeah. And that's probably why he started with Geodia then, because they definitely, if you have a lot of Geodia on deck and it's a lot of weather, you can become quite seasick
Yeah.
just from the smell.
You see some of these anemone are with the tentacles inwards. So those are the anemone.
The orange
These
balls.
here, those are anemone with tentacles. Inside the
cavity.
Yeah, it's the gastro.
Yeah, because
gastric
it's one
cavity.
cavity. They have just one.
Yeah.
It's one opening into the cavity, and that's where the food is processed.
Mhm. So they have a proper anus or
Well, it's the same as the mouth.
yeah.
In and out the same
Simplified.
place. Yeah. What's that long thing? Is that another carnivorous sponge?
Oh, I don't know. Probably this is something on top of sponge or something growing on top of this stalk. Could be carnivorous sponge. There's plenty of those around.
Okay, I will hit the button for carnivorous sponges as well, and for Rosellida. Another Lysodondoryx.
Oh, yeah.
I'm just waiting for you to tell me where my amphipods are, since I'm telling you where your sponge is.
Everywhere.
Everywhere. It's easy. So all of this, that looks like sedimentation. Do you think
brownish?
that?
I think it's yeah, the result of these pulids dying, and the tubes that they produce are like getting in there.
Mm-hmm. So all of the white dots you think are
Yeah.
serpulid?
Yeah, indeed.
Yeah.
To make a closer look.
The small white dots we're talking about now.
Yeah. There's plenty of those. And they're producing like a kind of a microhabitat where I'm pretty sure there will be many different small creatures living.
Yeah, I'm very sure about that too.
Yeah. There's some Asconema foliatum. There's another anemone there with the tentacles inwards. Most of them are like with the tentacles everted, trying to find food. When they get food, they get the food with the tentacles inside the cavity where the prey is going to be digested. Yeah, we're like going upwards to see whether we reach the very top of this mountain.
So the plan is to get to the top and then follow the ridge a little bit again like we did last time. But this wall is so pretty that right now I'm hoping that it will never end. It's also super interesting to see this community of animals together. So very rich. See there? Amphipod going down on its way to food.
So we don't need to forget that we have to take our eDNA samples when we reach the very top.
Because one of the things Sergi and I are doing here is, in addition to finding out everything about the sponges and specifically about Lizodendorix for Sergi, and trying to find out about the smaller animals for me, we are also trying to find out the environmental DNA, so the DNA that is left in water or sediment or filtered by natural filters such as some of the sponges from Sergi. We try to find out this so that this is a less invasive method, so it's still in a let's see if it really works stage, I would say. And one of the reasons it's still in a let's see if this really works stage is that we need to build a library to compare the environmental DNA with, so that we know which species we have in the environmental DNA, or eDNA as we like to call it. So we take samples of water and sediment at the same place so that we can compare as well. And then we find out which DNA is in this sediment and water, and we compare it to what we already have of libraries for species from this area, so DNA libraries for species from this area, and at the same time we're trying to build out that library further, because we don't have anything specific for the mid-ocean ridge or the Arctic mid-ocean ridge yet. We don't have anything even specific for this whole sea, this larger area of the Nordic seas. Many of the species that were found here historically have maybe not been refound, so we know of species names and some.
That's a very top.
That's
right?
the top, yeah. Some descriptions, but we don't know their DNA. So that's one of the reasons why we're collecting all of these animals. It might seem a bit obsessive to sample all of these things, but it is to make sure we have a good library of both which species we really find here, and sometimes we do need to look at them in the lab to decide properly which species this is, and also we try to then use these animals to, sometimes we need to make re-descriptions, sometimes we just need to identify it to the correct species and say, okay, now we get the DNA from this species, and sometimes, quite often maybe, it is an undescribed species, and then we need to describe it first. And modern descriptions, of course, normally now always include a DNA voucher as
Yeah.
well as what we say, morphological or a physical voucher at the museum. This is a beautiful rock.
Look, this is a pin around it.
Ooh, yeah. Sea spiders. Here we come.
Oh yeah. Okay.
So now we are
Yeah.
at the ridge, and it's a completely different fauna. We have
These small anemones. It's
small
soft corals, right?
anemones and soft corals, mainly soft corals, and then some Rosellida.
Those are not the same ones as the ones we've been.
No, these are some other ones. So we should maybe also try to get
Yeah.
a sample here.
Those are really small if you compare with the pinkish that we've been seeing in other areas.
And all of these long, thin things that stick out, are they all carnivorous sponges?
Yeah, indeed. So yeah, we need to. I think we should. Yeah, we should take a small sample, right? How about taking a rock, small rock? So we're gonna try to take a rock with this soft corals.
If we
Yeah.
can find a small enough rock that it's possible to bring in.
Oh yeah. I don't know how big is this one? What do you think? Is it too big?
Which one? Can you
12.
point to my screen here?
What do you think? Is it too big?
Yeah, no, okay.
Is that the one? Yeah, that's the one.
Yeah, it's a little bit big, I think. It's
or even
like-
a smaller one. Oh yeah,
yeah.
here. It's
Which one?
somewhere here, one of those. It's one or even here. Yeah. That's probably too big, yeah. But let's just first
Let's first
Enjoy
just
a little bit this.
enjoy a little bit and zoom in and look at some of these.
Can you turn off the list, please? Again, and this is the
Yeah.
Solenogaster.
The Solonogasters are everywhere. I have
Uh-huh.
never seen this size of Solonogasters before.
This is the orange, wormy-like specimen that you see just below the lasers right now. Well,
Which one do you want? Because we got quite an angle here, so.
I think this one. With the scope. Can you turn off the laser so that we can have like a still image, like a
Thank you.
I'm not going to turn it.
of this habitat? Interesting.
So I need to change position
Yeah.
somehow.
Yeah.
The other way around.
Ah, yeah. Tilted.
Yeah.
That's another one. These Solenogasters. Probably another one in here. It's plenty.
And there.
I mean, just seeing Solenogasters everywhere, right?
It's like when you're learning to
Much.
observe a new thing, then all of a sudden you see it everywhere. Is that the sea spider again?
Sea spider, yeah. There's quite a few of these.
There.
spider. You see?
Yeah.
Uh-huh.
Okay, so we are in the Solonogasters heaven.
Can you get it from that?
Pretty difficult.
So, okay.
Trying to position the
Not-
ROV. think it's going to be a really challenging taking samples from the sediment here because it's mostly rocky.
So,
Yeah.
Do you want to come further back?
just like, where is it on your screen again?
This one. This is one. That's the one you just pointed to. Yeah.
That in just in front of you.
Was this one you said?
That one here, this should be like
Right, close on the screen here?
yeah,
Yeah, that one, yeah.
for sure.
Do you want to come further back?
Well, the walk as well. Is it?
Yeah, the white one you
But I got
Scoop. Scoop.
I think
rock
this
on
could
my
be
left.
enough, right?
Oh, there's like a rock on my left.
I think the scoop would be the best, yes.
Yeah.
On the top there. And a rock there. Further back. Yeah. Back towards more left.
Aye, we're still 6 degrees here, a good bit off the run at the front. That came away. Try it and see if you're height. Try going down, I think it came
I can, yeah, I can come back.
Aye, but it's here.
Oh, right, yeah.
At the moment, the front's in here.
Oh, yeah, no, there you go.
From here. Want to move from here?
Yeah, just to see if we reach it.
So
Front's
one
in
of
here.
the difficult things with sampling is of course that the two manipulators that we have available need space and we need to sit at a good place. And the things we want to pick up are normally a little bit bigger than
Yeah.
we think they are. And look at that rock on the side there with all of the carnivorous sponges. That's crazy.
So you know where to use scoop?
The front, yeah.
And we are sitting still on the edge of a hill.
Scoop for this one, yeah?
Yeah.
Yes,
I think
please.
so.
So I'm hoping we're not tipping over. Gives me a little bit of dizziness to look down that hill.
So which ones are free? Top left.
I think
Yeah.
everything apart from
Top left.
I was going for the white peppers.
Do you want it in? Yeah, okay. That was a beautiful shrimp swimming by in a quite peculiar swimming method. So, if we are on the top now, are we also... No, it's the same transect, right?
Yep.
Yeah, okay. Okay, so while we're now at 1,445 meters depth, the water temperature is minus 0.4. So slightly warmer. Not really still swimming temperatures, no. But it's also slightly different salinity to previous. So it's obvious that we are in a little bit different water mass,
Yeah.
which is what we have been expecting since we are on the east side of the ridge and not anymore on the west side of the ridge.
Which one of those? Potentially also be this one if you want. So I cannot reach to this one with the camera now.
Okay.
If you want to go for it, then we'll be able to see the.
I mean, if you want that one, we can like
No, it's fine.
go back.
So this one is fine. I mean, the one that you think it's the easiest.
Yeah.
This one is closer, I think they are easiest.
The one
Yeah.
close.
These
Yeah.
ones.
Yeah.
way here, just
So yeah, and it fits in the scoop.
So what I am also seeing from our log is that we have
Does
the
it?
previous sample was exactly 1 hour ago, and we have moved 100 meters up the hill in that 1 hour, if you are wondering which speed we are moving at. So 100 meters in 60 minutes on a more or less vertical wall. I think Usain Bolt would have done it a little bit faster.
That was one of these soft coral, right?
Yeah.
Lion.
So yeah, this ground is very hard here.
Yeah.
I don't think we will try push core. here.
Not here.
This is not going to be the place.
Oh, on your elbow.
Is it attached to the other rock?
I think it's just a part of the bedrock, more or less.
If it's not loose, just leave it.
Yeah, don't try to take the whole mountain.
Yeah, we thought it was loose.
about this one.
Yeah.
It's too big. Probably it's
Ah, yeah, it can be right.
really big.
Somewhere in
The
the
balance of plants in the idea, the weight, pick up that rock.
Okay. Then
That's a drop in the
yeah, don't fall down now.
Should we already,
Let's go on.
It's too big, this one.
Yeah. Wow. Indeed. Yeah. Don't worry about that. We'll find another spot.
Yeah, starboard right.
Yep.
So we're gonna use the scoop now, but I'm pretty sure they will in the future.
Yeah. So these big white sponges in the background there, they are again the Rosellida?
Yeah, I think so.
Yeah?
This is as far as we can go for now. And
But
probably thanks to
this-
Leo and
yeah.
Paco.
but at this space they have not decorated themselves with the crinoids.
There's no crayons.
So they are not as fashion conscientious. Yeah?
Yeah, we haven't seen any, right?
No,
do
the
you
crinoids
recall?
are sort of missing here. But we do see some of the Ascidia every here and there. We see, and remember those Ascidia, they are our closest relatives in this area.
Look at this.
Yeah, look at that
Huge.
sea spider. He looks kind of tentative about the whole
Mhm.
thing. Slowly going around.
Yeah, I wouldn't say it's in a hurry, right?
No, I read one place that they are pushing the blood or the hemolymph around in their body using what is sort of more or less the equivalent to farts. So they are
Okay.
using gases from. So they're farting the blood around the body. Which might not be too polite to talk about, but it's Monday, so it's okay.
Absolutely.
The beginning of the week.
So we have these kind of quite large. We can run it.
Lasers on her.
Yeah. Oh, it's,
Oh yeah, look at that. So between those dots, my friends, it's 10 centimeters. And between, so that's 15, right? From tip of leg to tip of the other leg.
And hold on 1 second, because we want to talk about these again.
You want to talk
These
about the
mollusks.
Solenogaster,
Oh, I cannot go.
the worms,
further.
the mollusk, the
Yes,
wormy
yes.
mollusks that you see at the base of that soft coral there. You see an orange little worm thing
Wormy,
sticking
yeah.
around, and that's a mollusk.
Solenogaster. We collected 2 in the previous time.
Yes, so we already have some of them in the lab, and
Can continue life.
yeah, we will bring them. So, what is that? Is that a jelly? The red
I
thing?
think it's shrimp.
It's a shrimp
Backwards,
going
right?
very slowly backwards. Yes,
Uh-huh.
it is. It's just behaving like, oh, there it's behaving like a shrimp again.
So we couldn't sample these small soft corals.
Because they live on
Here,
2
but-
large rocks. And they're everywhere.
Plenty.
They're standing like little choir boys all along there in a nice little row.
Yeah. Mostly on the edge of each of the
Yeah.
rocks. Oh look, fish.
Ooh, a nice fish. So this morning we came over the sleeping ground of these fishes. They were all curled up sleeping. And we were thinking maybe that would be nice to do for us as well, just curl up and sleep. But there has not been a lot of rest since then, because we did sample a lot on the previous dive.
Yeah, we are on a shift.
And the lab is full of samples that need to be sorted and preserved and registered, so that when we bring them home we know every detail we can know about them, and we don't mess them up with anything else.
Yeah.
So when you see us making all of these, or hear us making all of these notes everywhere, it's because we need to know exactly where a sample came from, and so that we can correlate it to the video and to the information we have from the CTD that is on the RV. And yeah, all of this data will go into the databases that we have in the different museums of that, where all of this material will go to in the end.
Yeah.
And we will
need this
spread
data,
it
yeah.
between several different museums. Quite a lot of, actually all of the sponges, most of the sponges will
Most.
go to the Museum of Evolution in Uppsala. Some will
A part
go,
of them will also
go
to the
to-
National Museum of Natural Sciences in Madrid.
yeah, that's where you are working.
Yeah.
Yeah. And then the small other animals will be shared between the Senckenberg Nature Museum in Germany and the University Museum of Bergen in Norway. So when things are in museums, they are available for science, because they are then listed into these collections where it's possible to either come and visit and study the collections,
Yep.
or sometimes, maybe not so much lately, it's becoming difficult to send things. But it used to be possible to ask for loans as well. But shipping specimens is quite a complicated thing.
Oh, there's a fish there.
Is it a fish or is it just a shape? I have been wondering. I think it might be just a shape. No, it's a fish, you're
It
correct.
is,
Ooh, your eyes. I left my glasses in the lab.
bit
This is my
far, but
excuse.
yeah.
My glasses are not here. I don't have anything in front of my eyes. I don't see a thing. I kind of
Okay,
got a little bit
yeah.
there.
Underneath the fish. It's just
So-
center. Oh, sorry about that.
You were going to check how high this wall was, and it's the ridge, so it's a hill on the ridge.
There is no soft sediment we can use to put
Not right
the
here.
scores, right?
Let's see what's behind the hill on the ridge. But is that round thing a geodia?
The one here.
This
yeah,
one there?
Yeah, the one
In the
where
middle of the picture?
Yeah, the one with the lasers.
And
think so.
yeah, the lasers and the hole in the middle, the
Yeah.
osculum.
I think so. There's some fun shaped sponges. We collected a piece of it, of one similar organism during the first dive. And we need to identify, Julio Paco will shortly identify it to species level.
Yeah, Paco is our geodia hero.
Geographic.
And oh, look, there is again all of these going along the ridge.
Yeah.
Okay, would like to have a new move
everything.
direction north at 0.1.
So here is a combination of soft corals and carnivorous sponges and an enormous sponge of
Okay,
something.
thanks.
And that's a hexactinolite, right?
Not
Ned?
this one, I think.
No?
This is not.
Ooh,
the other ones
look at the
are
shape of that ridge we're going along.
probably not this one. Yeah, that's beautiful. These small soft corals.
But also look at the landscape. It's
Yeah.
crazy.
It's quite different from the previous dives in other areas.
Yeah. I am wondering how much pain it would be to go for a walk in this if it was above sea.
Yeah.
It would have been quite steep. And also, just a reminder, this is a seamount next to, on the side of this mountain ridge. But now we are on the east side. And this almost sheer wall going down to the west. So we're going towards the north now along the ridge of the mountain. And there is this wall to the west,
Yeah. Can you go a little bit faster?
which is quite
Sorry.
interesting.
Faster,
Yeah.
There is another fish hanging out there in amongst all of the soft corals.
Yeah.
And they just make the whole mountain look fussy.
It's not that they cover the entire rock. They just cover the part of the edges. That's
Yeah.
where they get more higher access to the water column.
But it's also interesting to see they're all facing east, so
Yeah.
the water must come from the east or from the south or something where we
Yeah.
are coming now. Because they are trying to get food from all the particles that are floating around in the water. And seamounts such like this are making a lot of little mini currents around them because the general current is coming from the south to the north in this area. And whenever there is a seamount or anything, it will make a little extra diversion for that current, so that they will have little eddies and little
Bridge,
curls and
ROV,
whirls and
can
stuff
we
like that.
speed
So
up to 0.2?
we will always have lots of extra little twirls and
Yeah.
Okay, thanks.
difficult to imagine beforehand currents on these seamounts. And this ridge is a little bit up and down, right?
Mm-hmm.
So we will do a little bit of a straight to the next hill.
So it's been 1 hour since we got here, right?
Yes,
Good.
1 hour, and we have been going up. Ooh, yeah, we got to the top, and now we're sort of more flattish.
So we like trying to get on track again with this transect that was it was Jenny that was thinking about this transect, right?
Yeah.
She was the one planning the way.
So we have
to
done
go.
the wall climb, and now we will follow the ridge a little bit. And we see the ridge stretching out in front of us with little sponges or big sponges acting almost like beacons in the deep here.
Bridge RV, can we increase again to 0.3? Okay, thanks.
Yeah, definitely different environment, despite being at similar depths that we have already explored in other areas. So yes, you never know when you are diving in a place.
Yeah, this
Whether
is
you're gonna find
completely unexplored, and it's not even having a name.
Mm.
So seamounts are getting names when the official maps from, I think Geographic is at least putting out one map with names. But then the different countries are also putting names to their seamounts. So this one, so far, is unnamed and unexplored until today. So now we're trying to do a little bit of exploration to see what it looks like.
Some sort of technical points in the
Quite a difficult one, this one, because it's got a little bit of ups and downs.
Yeah, and it's also because the background map that we have is not, the resolution is not super good, since this is quite unexplored. So, I mean, well, it is super good for being in the deep sea and everything, but it's not, I think it's 50 or
This is
30.
more muddy, isn't it?
Yeah, maybe we can get close and see.
Yeah, because at some point we would like to get these push cores and Niskin bottles and our sponge around. What are these sponges? Okay, these are fan-shaped sponges we could potentially grab. What do you think, Annalene?
If it is muddy, that would be a good place.
Do you think we could land here? Well, no.
Yes.
somewhere around here. So that to
And check if it's
possible.
possible to do a push core.
Let's
Let's first check if the bottom is
Yeah.
available for push coring. But it looks like it at this distance.
Yeah, pretty much. Let me check which. This is one of these.
It's a little bit rocky.
Yeah, to get closer, you see.
Yeah, that's the thing about getting closer. You always see more details when you're close. But we have the Serianthids in there, so it must be a little bit deep, right? So we have these tube anemone. That is a good indicator that there is a little bit of sediment. Yeah, let's try here. And make a feel for the possibility of a push core in between the cobbles.
Yeah.
Oh, look, there is do you think there is a burrow?
Yeah.
That's beautiful.
It might be the right spot.
So let's zoom in on a couple of things here.
Like this oriented, right? And,
Yeah, let's look at that one. And look at all those little orange
yeah,
dots.
Those are the
Anemone?
anemones. Yeah.
Little orange anemone dots. But then around them are these are sponges, right?
Granulas.
That's craniella.
I think these are the granulas.
The brain sponges. And then anemone.
Yeah.
This anemone lives down in the sediment and can subtract down in its tube when it does not like to be up on the surface anymore.
So we're gonna try to get some from the push cores, like somewhere, I think.
Yeah, somewhere there, right in front of the. Yeah. Can you just quickly zoom in on that thing that
Reach
might be a
LV,
burrow?
can we change the direction to 050? Okay, thanks.
Yeah. There's a barrel here.
But we don't see anybody at home. And it's covered by sponges.
Yeah.
That's a fancy house. And an opening of almost exactly 10 centimeters, huh? That's a big opening.
So let's just, sorry, I'll go down.
Now we are going to look at the push cores.
You also have the max, remember?
Yes, we do
They
have
were
a
not
max again.
used so far.
What was that push core number 5?
Happy anywhere?
Happy
Yeah.
anywhere in
Yeah.
this area.
Let's try. Not entirely sure whether this is soft. Really soft. Can you tell me what this is, please? It is soft. Indeed. Soft. Thank you so much. Yeah. Got it.
Okay. And that's number 5, isn't it?
I think so.
So these samples will be for environmental DNA or eDNA.
Yeah.
And it's quite
Great.
muddy when we get under the rocks.
It's number 5, right?
Yes.
Just this one. Yeah, I think it's fine.
Two push course here.
Yeah.
2 push cars, please, yes.
Yeah, 2 replicates in here.
I think we have both coffee cups left.
Same kind of area as well.
Yeah.
Yeah.
Not super close.
Here, okay?
Yeah, that sounds perfect.
Oh, the anemone was like getting disturbed.
Yeah, it
Yep.
Alright.
dove down into its hole.
Bridge, LD, can we stop for now?
That's important.
Bridge, RV,
Yay.
So 2 push chorus.
can we stop for now?
Stop.
Okay, thanks.
Now we need to do the Niskins.
Shall we do also the marks?
Let's do one more here as well.
Dort, we're gonna have flags here too. The mugs.
Maximize.
Yeah, but let's do the Niskins.
Yeah.
Can you trigger them, the Niskins?
This can, yeah. Yeah, this can.
Can I? So the bottom one. Yeah, worked.
Bottom.
Top one. Looks right.
At least the bottom is
The
okay,
bottom
right?
worked.
Yes.
Yeah.
This one,
Top one, the top plug's definitely, and the bottom one should be it. Just take a look, 2 push course.
So we will use the bottom one on this.
Yeah.
So the ROV has 2 Niskin bottles, which are like bottles that can be closed at the depth that you want.
So
And
it's
then-
like a tube that is open at both ends until we close it. So we have the water from the
This exact point.
yes,
Yes.
and then we just close the top and the bottom of the tube, and then we have how many liters?
I think it's like 6 liters.
Yeah. So then we
something
have
like
a
that.
6-liter sample of the water right where we are when we trigger the Niskin.
Mhm.
So that's a way to get the water samples, and both water samples and the push cores are going to be used for the eDNA.
Mhm.
And now comes our cutest sampler. That is the coffee mug.
Mhm.
it would be quite difficult to drink coffee out of this because, as you can see, there is a net at the bottom of the cup instead of a bottom at the cup bottom. So we use it to sample sediment into the
Close to the
bank.
area where you also did
What's that?
not the same ones,
but
close.
close to,
Close to where you did the push course, but not.
not on top of it.
Oh, yeah, I'm sorry. I think you are right on top of one of the push core. Ah, yeah, that one.
Yeah.
A little bit further if you want. Yeah, yeah. Like here. Yeah, that will be good. Oh, there's a rock or something. We can go somewhere else.
To go back to the door. Try and do it.
God bless you. Yeah, the idea is that we're gonna have a small representation of the sediment in this area. It's gonna be filtered, and hopefully we'll have a bunch of
Amphipods.
amphipods or other creatures that live in the.
Yeah, we want other creatures in addition to amphipods. I'm not going to be against other animals. We want them all.
Yeah.
But so far in these samples we have found lots of little worms, a few crustaceans,
You can take a little bit more time if you want to clean it up.
and some mollusks. These are the main groups we have found in these samples.
Yeah.
Of course, we're seeing on the soft sediment, we see the seriantids, these anemone that are pulling down into their tubes. And we see sometimes, but not here, we have seen stalked both crinoids and stalked sponges, but right here we don't see them.
Nope.
So this is a different
Quite different, I would say.
fauna, yeah.
Some different species.
Oh yeah, look at that sediment sample. That's gonna be a joy in the lab.
Latte, that looks like, doesn't it?
Are we all dreaming about an espresso right now? Or is this a tea gang? You're the coffee gang.
I am, yes.
Yeah, we have the coffee gang. Oh, and we have one tea man back
Just a
there.
latte or an Irish coffee. I like Irish
Yeah, it's quite thick. It's nearly going to.
Oh yeah, it's fine.
Yeah,
You can just put it back into.
it's definitely an
Firebox.
espresso, a double espresso.
So it was top right. Right.
Yes.
So it's gonna be a little bit
Oh,
of
it's
work
a-
in here.
Yeah,
to be
that's a
done.
big sample. It's gonna be, but 11, 12. We only have 2 hours left of our shift, so this is work for the other group.
To keep them busy, right?
Yay! To keep the pressure up.
Yeah, we need to have some more samples. Yeah,
Yeah.
and they will be happy to be working on this material. Oops, there's a
So one thing you can easily see here is how strong those manipulators are, those arms. They are very strong. Do we think we still can sneak down the coffee mug? Yes.
yeah.
They're also very good at doing very detailed work.
Do you think we should take another one?
Not here.
Not here. Okay. Thank you guys.
Thank you so much.
Then ideally, can you?
Did you want to do a second coffee mug?
I think we will do a second coffee mug another place so
Another
that they
place,
will
okay.
look a little bit different maybe.
Okay.
So
At
ideally
least that
now,
will be the idea.
turn a little bit to the left because there were some sponges. You want
Right,
to grab
okay.
a piece of sponge?
You're going to grab some sponge,
Yeah.
okay. Seems to offer job. Right, sorry. And you're going to grab a sponge. Yeah.
And this sponge we will use as natural filter
Mm-hmm.
also for the eDNA. So
Yep.
that's why we want the sponge from close by here so that we have all our eDNA samples from the same place.
Mm-hmm.
That makes for the
Do you want to spend
best
it to grab it?
setup.
No, that's fine.
Yeah, you can.
Look at this.
Where did we have the sponges?
I think they were like on the left. These sharp fan-shaped sponges.
Oh yeah, you want the fan-shaped sponges for this. So Sergio has been working quite a lot with finding eDNA from sponges,
Mm-hmm.
from natural filters.
Deep water, shallow water, and sponges they perform really good at collecting the DNA.
Yeah, well,
Of the
they're
surrounding
here all the
habitat.
time filtering, so
Mm-hmm.
something is, now you have to help them find your sponges.
Where do you want me to go for this one?
Yeah, just go up and tilt to the left. No, not these ones. There's a few in here. So
I think we have to lift up and turn
Yeah.
around a little bit.
Right, okay.
Because is that a Claudia?
yeah, but the Geodias are not the best at filtering. Sorry. So there are two main groups of sponges: the low microbial abundant species and the high microbial abundant species. High microbial abundant species as Geodia, they can be like more than 90% of the tissue of the sponge filled by bacteria archaea, which is like a crazy amount of microbes in there.
So they are an ecosystem in themselves.
Whereas the low microbial abundant species, these fan-shaped sponges or Lislandorix, Complicata, and many other ones, they have like a less amount of bacteria and archaea, and they are like more active filter feeders. So this makes a difference because the Geodia actually is not filtering as much as other.
Oh, because it's
species
just
do.
hanging out with its bacteria instead.
They rely more on the bacteria. This is a symbiosis with the sponge, and they get food from the bacteria. Whereas the other ones, they have to filter more actively.
So is there an evolutionary, did they split up and then one side became
High
more-
and low. Yeah, that's a good question. I don't think so. But there's like
So it's
some
something that
sponges
has
are-
happened several times
Yeah,
in
Sorry,
both.
sorry, sorry.
I'm looking for sponges.
Oh,
I've
so
got
Sergi,
I think
Sergi, you
there's,
have to help them look for sponges.
So if you fly a little bit up.
Yeah, no, I can fly up. Oh, so.
He has been moving around. Do you see any? If you
Not
zoom
really.
out.
I think they were like closer to the edge on the left hand side.
we are on the edge.
Yeah.
So I don't think we can do a lot more.
I think they were a little bit backwards. You can turn my head to the left. Yeah, to the left. I'm probably a little bit backwards.
There's a list of Benthics there, isn't there? Because then we would be able to use that one, right?
Yeah, if it's a list of Benthics, yeah. That'll be
And now we have a dust cloud.
There's something
Yep.
there, but what's that?
Is that any good?
Not sure about it. I think this is one of these Asconema, right?
I think so. Are you asking me about sponges now? Yes! I'm at the level where I'm being asked about sponges.
This is not the most interesting one.
Okay.
If you turn a little bit and go backwards, because this is an exactinelid, and they don't
They are glass sponges.
Yeah.
They're made out of glass.
filter that much as
They're just standing there being pretty and made out of glass,
yeah,
huh?
that's it.
Ooh, don't touch me. I'm made out of glass. I don't need to filter. No, but they're super pretty.
Yeah, they are.
And very interesting that animals can be made entirely out of glass.
Almost 10 location now. 0.8.
Turn my head exactly.
Well, now we're really into the cloud, huh?
Yeah.
So this is the thing about being in a soft sediment when you're looking around. We do stir up a little bit of the sediment.
I've got to watch my turns or something.
To the time.
We don't want to get knots on our umbilical, right?
No, we don't.
Yeah.
That would be very sad. So of course the umbilical or the cable that goes from the ROV and all the way up to the ship
I see something. Yeah.
is very, very important to take good care of. That's our
That
lifeline
bunch
to
that I was
from the ROV to the. That's where all of the communication with the ROV is going.
I think this is the one.
Both the pictures that we're seeing and all of the information that the pilots are giving the ROV about where it should go and how it should
Should do
behave.
the trick.
So it's an amazing piece of electronic hardware.
Is that a sponge or a rock?
I'm not
Thinking
sure
the same.
anymore.
It's a rock.
It's a rock.
Rocky sponge.
Okay, let's go and see if we can find something that is
Yeah.
close.
Otherwise,
Because-
yeah, it's.
Yeah, we're just kicking up.
Yeah, I think we just have to try to see if we can get somewhere
Yeah,
out
we'll
of the
have
cloud.
another one close. I mean, it's
Yeah.
the same habitat and.
Just some moving forward. yeah.
Sorry about that.
No, that's okay. And then we get up to this.
Yeah, let's just get out from this.
Out of the sandstorm and hopefully without any bows and ties on the. There is another spider.
Yeah.
Yeah. And look, it's another hill right in front of us,
Uh-huh.
like
If it was
a wall. And
These are
Sergei
Asconema.
immediately goes for looking at the sponges on the wall.
Asconema. Yeah, I think it's Asconema foliatum.
Anything, yeah?
Yeah.
It's the wrong type of sponges.
I mean, we could
So now
potentially
you're
grab it.
picky as well.
Yeah.
Yeah. All sponges are equal, but some are more equal.
Mhm. Yeah, we're not finding the right one. Yeah, these sediment all over the place should be cleared. Yeah, currents are gonna do the job.
So this is definitely a wall again,
The
right?
wall, yeah. Just beside the area where we're collecting the samples, the eDNA samples. Yeah.
But we made the whole area quite sandy.
Mm-hmm.
I think that one is the one that we were coming from. That wall was the one that we were coming from with the rivers.
Just trying to find the right sponge in here.
Down. We were going down.
Oh, you think we are going back to the same place?
This wall, I think so.
Uh-huh.
Because we were going down and then we just move here and then we are.
Oh, then we need to actually sort of go the other way.
Yeah. That's nice.
Yeah. Gotta be patient.
I haven't made it basically because we are moving to the same direction. I think
Oh yeah, those are
Side turns.
just that.
we go clockwise.
Oh yeah, no, close.
Yeah, those could be
Yeah,
a potential target.
Me to turn my head all the way around.
I think so because we were coming from here, then we was looping out, we are coming this way. So
This face.
Yeah, all of a sudden it's all of the sponges you don't want, right?
Yeah, exactly, Nelly.
should be roughly 0.7, wasn't it?
So if you spot one of these fan-shaped ones, just give us a shout. Is it the one?
The thing is that
Oh, that's a big
we
one.
need to go the other way because we are coming again.
But I don't you're saying that we are going the wrong way.
Yeah.
Reach RV, can we go ahead at 0.2?
Just wait until it gets
Yeah.
on track.
Okay, that's it, thanks.
So now there are so many people in here. Temperature is going up.
Yeah,
It's
because the
not me.
shift of the
Of the
pilots.
pilots.
So meaning that it's lunchtime.
Oh right, yeah. Yes, no, but taking them out.
Halfway long.
I pretty much I don't know, it's a halfway. Nice.
We just head up. Yeah, I'm just coming back. Yeah.
So how long until 17 minutes?
Yes.
Till change of the tape.
Are you hungry?
A little bit, yeah.
So we are around lunchtime on the boat and around shift time for very many of the crew. Both the ROV pilot crew and quite a lot of the crew on the boat as well. Shifts around the meals.
How are you going?
Yes.
Just ahead, just we got up here.
We ask ahead means doing northeast.
Then we can left. Go left.
We ask to do
Well, I'll
We're trying to get again to the transect.
Yeah, but we are just turning around, and it's easier to turn around if you're a little bit up, especially when we're changing everybody flying as well, and it's good to be either sitting on the
Can we
ground
have
or...
a change of bearing to 180 at 0.2, please?
So this is the reason why you're seeing this blue screen now.
Yeah, no, it's north we're going, isn't it?
Yeah, we're going north.
So why is it
Southwest there.
Yeah, because we went down, we should go up or the other. We did a loop there, and then just coming down, in the middle of the first, and now we should go north.
That was the ridge up here at top.
Yeah.
I don't look at that, you just go back and right.
back to left, yeah.
Oh no, in the lateral left, go back to forwards.
I can't see.
Yeah.
Put the range and the solar there, so that
I think we confused them a little bit with all the turning around.
Yeah. All these
Searching
sediment.
for the...
Let's push forward.
Yeah.
Yeah, best thing would have been to go to continue the transect and then get proper response.
Yeah, you're pushing forward.
Yeah, just
You're going. So from the southwest, to bring your heating. You're heating on clockwise here.
clockwise we'll go. Because we came counterclockwise to go left to look for those.
90 degrees.
Yeah.
The ridge is going to you're way off the ridge here.
Okay.
The west there's keep going to eat. See what changes. Just forward, you know.
Yeah, just forward, yeah.
I see you're still going. You're going northwest there. So bring it another 45.
So we are 40 minutes away from the ticket now.
Yeah. We took a little bit up, and then we will go down again and find the seafloor for you all. No worries. The transit will continue.
Yeah.
So that's a ridge here.
That's at 50 meters range. So bring that down as you go along.
Now I see the seabed.
Yay! Back to the seabed. We like the seabed the best. Yeah. But now we can see all of this marine snow in the water.
There's a wall.
Vessels coming
Yeah.
south, isn't it, Andrea?
Yes.
But
It continues
crossing over for a distance?
No.
Continuous,
Just
continuous.
Continuous, okay.
Yeah,
I
we're
can see
coming
now.
right up until this wall.
That's 0.2.
Mhm.
So how do we end up to the southwest?
Because we go and we pick that, let's go left to look for to come round, to go backwards,
So
to
now
look
it's
for
centimeters
sponges.
north.
Okay.
That's where we land, we'll
Yeah.
pick that, so go
now
and
it
behind
goes
us. That's where
Going up.
So
Yeah, yeah.
Yeah, that was us.
Mhm.
That was our fault.
that's where
But look at this. We have
it's off to the left.
the same wall
Yeah, similar as
fauna again.
previous
It's a
one.
very interesting combination of everything.
But I'm going what he said.
Need to mark the lasers.
Lasers on?
Yeah, thanks.
Ask me, and I'll give him a fucking answer. Right, no fucking ask.
Alright, that's alright.
And our next shift is coming in as well. So I hope they have already had lunch.
Yes.
Yes.
Noodles.
Yay. Comfort food.
I'm not sure.
So we have done a lot of we've done the push course and the whiskey and one coffee mug. And did you see we tried to get the Slaughtersaurus? It got away. Free. And then we also this we didn't get. This was a try to get the small soft corals.
Yeah.
The ones that are on the edges of everything, but they're
Spend
always
some
on the
time.
edges of big things. So Sergio wanted us to just pick up the mountain, and that didn't work.
Uh-huh.
You always
but-
wanted
there was
that.
something
That was
lying.
interesting.
On a bubble.
But that's the jelly.
Oh, yeah.
That was a nice jelly.
So the idea, guys, was to once on the top try to get one of these demo sponge. Piece of demo sponge. There's not that many.
Demo sponge. Demo sponge is
Yeah, like not exactly
any demo sponge.
Yeah,
because
because
yeah, it's
he
for
wants
combining the mud. I mean the sediment push core and the skin with some species, with at least one species that filters a lot. Because exactly, and it's they tend not to.
But then shouldn't we take jelly?
Geodia, for sure not, because it's a high prevalence species. So I think one of these fan-shaped ones may have, yeah. We saw, yeah, we saw
so
some
video type change in 10 minutes.
many.
Options which are
Lysidendorix would be nice.
So if you find one Lysidendorix, just
That's zoom in on that. That's squid.
Let's go with something.
oh wow.
Wait. Could we stop?
Yeah. With a fish.
With the fish?
Can you go? Can you try to chase this
No, don't go.
a little bit? Get closer.
It's a squid with the fish.
Oh yeah.
Try not to move the camera, then I'll go.
Oh sorry. Look at that. Oh.
Yeah, he is hungry. Yeah. Not that way. That way.
We need the whale eating the squid.
But that was nice.
Yeah, alright, sound. Right.
Can we take the lasers off?
Yeah.
Lasers off.
There you go. Thrust the wash.
It's getting out from the still there.
I just wonder how he's going to manage to eat that whole thing. He's going to be full for a week.
that carrying eggs, you think?
Mm.
That is eating, catching a fish.
Lovely.
So it was a squid having caught a fish.
You're up there.
Yeah.
Nice.
Hunter.
Nice one.
Yes.
Yeah. So I'm following this
So hold on. 2 more minutes.
8.
Where's it going? Coming over the side?
Is that one of these fan shape?
No.
Yeah.
Is
Is
it?
this fan shape?
Yes.
Can you stop here, please?
Yeah.
Yeah.
Because we're gonna grab one of these.
One to the right at least, one to the left.
That one, sure.
So finally, Sergi, right before he's leaving for lunch, found his fan-shaped sponge that does not have a lot of bacteria and filters like crazy, and will be his
That is the one.
active natural
After 20 minutes.
filter for the eDNA.
Yeah.
And it's good because then we will have a happy Sergi going back to the lab.
Yeah.
And we aim for
We're
happiness
going-
in
yeah,
all
we're
our
going-
people
we're
here.
going down slope, so the back of the RV is hitting the cliff.
Okay.
If I go forward, I'll go over it. So
Uh-huh.
this is
Can you line it here?
no, because it's going down that way. I can swing around
Yeah.
and face up.
Yep.
Yeah.
Like there.
Yeah.
Should be.
And now he's
Oh, yeah, now I
redirecting
see
the
that.
Yeah.
the entire ROV to go around to get his fan-shaped sponge without much bacterial
Yeah.
action.
And have you had a good catch of Echinoderms?
Not really. We didn't do
We
many.
haven't done any Lizardendorix.
So we
But
were
we've had some before.
four.
Should that be fine? But the one that we took
If
on the
you
previous
took, if you take a few more, that would be good. The thing is that we don't see many in this plateau area. So they were more like not up in the wall, but just a medium depth, probably.
Okay.
And we want another couple.
You think you
Yes.
can reach that one?
Probably to find
Should
out what to grab or slurp.
Uh,
Yeah.
and see the beautiful spider right
Yeah.
next to it.
So I'm going uphill there. I'm going uphill.
And then we found out that
Yeah.
these little orange dots in the foreground are also sponges, right? I think it's pretty.
Mm-hmm.
Well, I mean, just aesthetically pretty.
Yes, okay. Yeah. Slurping. Yeah.
So it's 5 minutes.
Okay.
5 minutes until tape change.
Mm-hmm.
Oh yeah, 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 yeah.
Ready to go?
Yep, ready. The one behind
So
you.
one on the back.
One behind,
Yeah.
Up it. Is it?
Yep.
Yes.
That one.
Can you zoom in say before because I'm not so sure about the idea.
Do we need the lasers on?
Huh?
So you have a...
I just want to define the sponge. Make sure it's not an insect in it.
You think it's an exactly?
Yesterday we collected insect in it, similar to that one. You see the skin? You see the skin?
Uh-huh.
I'm not saying it is, but it's better always to zoom in before collecting some.
Yeah.
Oh, there's a
You see the
little
skin?
jelly.
Not really a demo sponge.
Well, we got a quite large part of that sponge
So
now.
that is it.
Where am I going with this?
Yeah.
Yes, please. We can put it in
Firebox.
the which is easiest for you. There's space in both.
Have to be
To the right.
Okay, you want to have it in the
The one on
port?
the right there.
In the port, both in the back are free.
Okay. So we do
But this one has the mark.
Yeah, no, but that's in the front. So the
Yeah,
front,
It's gonna be a little bit.
yeah.
It's fully out.
It was broken, right? Already? Had like
Can we
a.
stop the movement for now, please?
You will see in the images. So this part is started okay. You see? It's not a complete fan. It was just broken.
Is that reminds me of the one that they collected in shoes? The one that is right?
It could be. But I think this shape at least has more power of filtering, right?
No, you want
Potential.
the most spongy.
Yeah. There's no other than that, right?
Yeah, we will find one.
If you find Echinoderm for sure, it's,
What are you doing to score here?
we did everything already. All the push cores and these in bottles.
So it's just about
And one mug.
So you can do one more mug and you can do Echinoderms, and there is quite a lot of space still. We have not slurped anything, so there's 2 slurps left for you.
How far are you in the concept?
Not very far.
Okay, we're not very far.
which mark is left?
The front left. Okay, so it goes in 4 instead.
So it's gonna be EV4.
Yes.
Thank you so much, guys.
Okay, 1 minute until tape change.
And 1 minute devoted to this Echinoderm.
Yes, 1 minute devoted to the beautiful Pycnogonid.
Ooh, look at this beauty.
Hey. And to the
Oh, carnivorous sponges.
carnivorous
Oh,
sponges on the side. Fancy. And then we were agreeing that these were tiny anemone.
Mm-hmm.
The little brown,
tentacles in.
yeah, the little brown orange thingies are anemone. But these things up here, they are sponges sitting on the orange.
Or anemone. Could be, right?
Yeah. You
Yeah,
think they're
anemone with
anemone
that.
still, but sitting on the,
Yeah, because there's
they're sitting on the sponges, right?
Yes. It's really
There is a beautiful specimen.
Now it's coming.
Frank again.
Okay, it's going to come and check
On
out
the
the.
run.
Okay, please change the tape.
1, 2, 3.
Stop recording. Thank you.
Yep. Okay, guys.
I have an empty one for you.
Okay. Thank you.
Beautiful shot.
Ready to start?
Yes.
1, 2, 3.
Start
Yeah,
recording.
anemone.
Yeah. Okay, then Anna Helene and Sergi are giving over the microphone to Franci and Julio.
Yeah, let's go for lunch.
So the next group will take over then. Yes. Okay.
Thank you so much.
Thank you guys.
Okay. Yeah, you're welcome. Yeah.
Hello, hello. Julio and Franci are back again.
Belly full.
Yeah. Exactly. Some nice soup. Okay, I give this to you.
Yes. Thank you.
Right, what am I doing? Am I going just straight up? Or back on the same?
So cool, these spiders.
Yeah.
Speak up. I like the
Their
spiders.
mouth. But these ones, a little bit more.
Yeah, maybe these are a bit more.
Their mouthpiece is really cool. This elongated trunk.
Could you put the laser on? Thank you.
Okay, let's see what else we can discover here at the unnamed seamount. Many more of the shrimp friends.
Yeah.
So one mark,
Yeah.
zombie and it's next. That's
Mhm.
it, right?
Yep. Ooh, there are a lot of sponges coming up.
Yeah. How about stopping on this
Oh.
area?
Hey, this grows very interestingly.
It's tricky.
Point 8 with a turn.
Looks like foam.
Point 8 with a turn.
I'd like to check those big ones.
Yeah, what are they? These ones?
Yeah, I think that is the same that they. What's the feature over there? They collect
yeah. Without squid.
on the first dive.
I'm just going to come up a
I-
little. I'm just going to come up a little bit.
Yeah, no,
So I
problem,
can
no
turn.
problem, no problem. Sure.
Right, because it's going to be a big turnaround, isn't it?
Don't do it if it's
Yeah, it's not so easy navigating on this terrain.
I'll put a big turn in the
So
other.
our pilots have to figure it out a little bit.
He's got to get that down, otherwise we could twist into the orbit.
What goes to that down?
So we're making a bit of a turnaround now. I already lost my directions.
Yeah, we are spinning.
Sorry. I just need this.
Ooh, okay, now we have a nice view.
Okay, yours.
Do you think it's the one that we just sampled, or Sergi and Anna-Elena sampled?
It's a bit hard to say.
It's a really cool structure. Is it even, would you still call this fan-shaped? Yeah, right?
Yeah?
Yeah.
Yeah, yeah. Well, I mean, those are just names, you know, so sometimes the shape is not that clear.
No.
And it's a transition between, it's something in the middle, but yeah. Yeah, it's a fun shape. Lamellar also, sometimes fun shape. It's more when you have a clear center at the basis.
Yeah.
This is more like lamella because you don't have a one point where the fibers converge, but
Lamella and-
many attachment points. See what I mean? It's like a wall.
Yeah, yeah.
So, but because they are sponges, they change the shape so much. That it's always just to have a reference. But yeah, you could call that reference shape. I would say that this is the same that they just collect.
Yeah, looks
So,
from the inside structure also.
Yeah, I will not collect this one,
No,
but I would like to have a nice look at it.
Yeah.
So to see the
community around.
well, to see
how
the
it
morphology
grows.
of
Yeah.
it, and maybe we should call it a little piece. The thing is that if it's a new species, you want to
That's
have
good to
a
have
nice
more.
view of the one that you are going to sequence and deposit. I'm just afraid I don't want to break it. It seems like it's a little bit.
Yeah, it looked as if it's very.
It's not that easy to break, you know, because.
Yeah, to break a small piece of. You'd
yeah.
rather get the whole thing than.
So, I don't know.
Do you have an idea which family?
No,
It is?
no, not
No.
yet, not yet. Not yet.
It's really cool. Also vein-like structures, not only the pores but also these. Yeah.
Yeah, well,
veins.
those are the fibers that give the structure and the like the internal architecture of
Yeah.
the whole body. So,
And it's growing on top of geodia.
it's rock.
Okay.
This is
Yeah.
rock.
Yeah. Not the furry geodia sponge.
I'm not sure if Paco is over there, but
I think he's busy with another school.
okay. I feel fancy that I could try this with the hand. I just need a piece. I don't need.
Yeah, might be a bit tricky to get it off, but you can try.
How big is that?
Where the lasers 10. It's over a meter, 1.5 meters maybe.
Could we turn the laser off for a second? Thank you so much. Another one. Yeah. This is nice also in the
Yeah, in the back.
background.
Yeah. Some small corals and sponges and anemones. Do you think maybe here at this junction it's easier to break it off? I mean, then it's a.
This is very big, huh? I don't need that much.
Yeah.
I don't need that much actually, but yeah, we can try. I mean,
What's this? Is it a carnivorous?
that's coral, I think. Okay, no.
See the polyps, right? Yeah.
Yeah, those are. That's coral. These are polyps. Let me just check that rock over there. It's just kind of beautiful.
And worms? What, like tubes of the worms?
Sure.
Oh, worm guy Sergi is not there anymore.
So I would try to pick a piece of the large one. In here.
I would think it's, yeah,
It's
I mean, this
easier.
is much bigger, but I would suspect that that's easier to break, even if the piece is bigger than the one that we want, because it seems more loose.
Okay.
But
Okay, let's try that one.
yeah,
Can we try to take this piece?
Big piece on the left.
Yes.
if it's a smaller piece, even better. But we suspect maybe it's breaking here. But if you manage to break even a smaller piece, even better. But might be hard to break it
Yeah,
off
okay.
because of its structure.
It's kind of a fibrous texture. So
That's one place with Rory. There, it's a bit better big.
Where would you like? Might not be able to fit in the starboard side.
So starboard is not available.
Might have to reposition. Yeah.
But this is in there, so we could put it there.
Yeah. So
Okay.
port back left is empty. Is available.
Yeah. Pretty big to get in.
It's too big.
See what comes.
Might be able to get it.
I think we should. Yeah. A little zoom.
Zoom in.
Yeah. Tilt up. This is fairly hard.
Coming
Maybe if you just
Yeah,
Snap.
I'm just touching it, so look, it's also fibrous.
Yeah,
Yeah, that's
that's
the one.
already. Very fluffy.
A little bit more zoom. That's it. Yeah, thanks.
sorry. More, eh?
Enough? Not too much.
Perfect. Yeah,
That's
Is that? Yeah.
really nice. Do you want to report?
We try port, yeah.
a reminder.
Yeah.
Could we slurp this up now?
Slurp, then everything will break, I believe, because it's too,
But
it's
if
too
we put it,
fibrous.
and if we put it
You
in,
want to slurp the piece, the little piece? Yeah, that's fine. That's
because
fine, no
otherwise it
problem.
might flow off, no?
Yeah, that's fine. Yeah.
Okay, close the drawer and slurp this piece.
Good one. Yeah.
Yeah, I thought you wanted to slurp.
Yeah, so,
So this is cast 11, right?
Yes.
before you do the hand drink,
Yes.
you get the slurp tank and you're
Do you want? Oh,
So in
4.
4.
Yeah.
It's okay.
It's okay to go with 4, yeah? Okay, so zoom in a little.
Sorry.
suctionality too.
Good.
Yeah.
Okay.
Resume their folio.
That?
That's fine. Yeah.
Coming from that way.
Where are we? Just no constant. You good there? Yeah, go in. Okay. So suction on that?
Yeah.
You're on, yeah?
Yeah.
Okay. I'm gonna open the jaws now.
Ah,
So
perfect.
much.
Nice.
Okay.
Thank you. Well, that was some nice sampling here.
That's it for sampling, yeah?
Yes.
Okay.
Thank you.
You want to follow?
I can follow the line.
Yeah, 1 second, I'll get the arms away. So get Andrea first.
So we collected a piece of this sponge that we don't know yet what it is, and to find out later on what spicules it has, and in general what more we can find out. Will you analyze the molecular genetics as well of this?
Yeah. DNA. DNA and morphology. So hopefully we can put a name on it.
Yep.
It's important because it's building an habitat. So when they are that big, we really want to know what they are.
Yeah. Yeah, they're super big.
Should I move the camera?
No, you're okay. Oh yeah, you can
We
square up
don't
your
have
camera.
a Solenogaster here, right?
Put your camera in the middle. Okay, Rob.
I just wondered if there was a Solenogaster here by chance, but I don't see any. So
No, I haven't seen any. So we need one mark, right?
yeah, we want one more mug with sediment. And we have another slurp chamber. And a lot of space on the starboard side for listening dogs.
How far we are on the transit?
Apparently not very far. Can try to understand.
Is it going up?
Understand.
Is it going up or is the summit? Is it going up or is the summit? It's going up at the beginning and then summit. Yeah. So we may be here at the end. So this is 1,451 meters.
1,051.
451.
The flow,
Yeah, so maybe we're
we
here.
are out here.
Or we are here again. But no, it doesn't look like it.
Yeah, maybe.
Yeah, that's what's
Okay, guys, if you're ready to move.
Okay. So I'm gonna put a half turn in.
Yes, we were looking in our smart folder here, where we have all our different dives nicely laid out. with a map and with a profile of the different depths. And we're now on the third transect for this site. We're at the Unnamed seamount, far up north, along Carnivorous Ridge actually. We're not on Mons Ridge anymore.
Oh, okay, yeah, that's a good point.
Yeah.
Didn't thought about that.
Yeah, I mean, neither somehow. We just, had another dive tonight, or last night, at this site.
Super flat there, those rocks.
Yeah,
Probably. Yes, that interesting. Those
where
rocks
we
are
explored
super
another
flat.
side of this
The rocks
seamount.
were super flat.
Also, a slope. We were climbing up a slope, and now we're climbing up another slope. And yeah, it's really interesting to see the communities here in comparison to what we've seen before further south. And similar depths though. And in general to explore a site that has not been visited before or not been explored and studied before. Yeah. For sure, it seems that there's a lot of biomass in general, biodiversity, and it also seems to be a very productive area. I've seen from small organisms up to big ones that there's a habitat built here, a lot of shrimp and filter feeding organisms, then some squid we've already seen, also in action eating, and how this whole food chain builds up here. Yeah.
Could we turn the laser on?
Yes.
Thank you.
Now it's Mallorca time here. It's t-shirt weather. And I'm all dressed up here.
Are you cold?
No,
I think it's okay now.
Yeah. But I don't feel like sitting in a t-shirt.
It's not Mallorca weather at all.
Julio is from Mallorca, that's why
Yeah,
we talk about Mallorca weather.
a bit warmer there.
Okay?
A bit warmer there.
Yeah.
Algo más caluroso.
It seems to be more even now. Or going down even, I mean.
So, could we go a little bit more port?
Yeah.
Just to
Towards
follow
the
this
edge, yeah.
crest.
Are those sponges Roselidae?
That seems to me a mix of Roselidae and what we just collect.
Ah, yeah.
But I'm not sure, actually. See the large one on the rock is what we just collect. Almost sure. Then you have some Roselidae.
Yeah.
You have some carnivorous sponge. So somebody is feeding off those sponges. You see the holes on those sponges?
Yep. Maybe
That
we had an hungry
seems like spongivore.
spongivore.
Yeah. And you see the bald rock is completely covered. Well, completely covered. It's covered by those anemones.
And what edge is here?
That's the rock.
Yeah, but it seems like these outlines of it. Is it just sediment on it? That seem to have like an encrusting outline.
All these large little trees are also sponges. Those are carnivorous sponges. Different species you can see here. This one that is more branching, and another one that's more like one single stem.
And this is the more branching one is the Asbestopluma, and the other one.
I'm not
Like
sure.
supposed
It could be
potentially.
Asbestopluma as well. Could be
Acostatus too.
Of the second one?
No,
as well?
no, of these ones.
No, this one, yeah,
Yeah.
for sure. This is nice. Just stay with them because something better.
All those in here in the back.
Yeah, that's the same
The
one
fan.
that we just
Yeah.
collected.
What's there in the back? Is it a fish? Or is it a squid? It's a squid.
Yeah, it's like your
Yeah.
It's more like a Ctenophora to me.
Oh, yeah, I was, yes. I had the wrong, I thought it was much further away. I didn't have the 3D comparison. It was like a optical illusion. Yeah.
Geographic. We have a movement bearing 005 at 0.2 knots. Please continue with me.
5 at 0.2.
Thank you.
There's a skate over there to the right.
Where?
There.
Oh, there in the back. Oh, yeah. Nice. We haven't seen skates yet on this dive, I think.
Or even on this area.
Yeah. That makes sense, since it's very productive. Like,
I'm not sure.
no? Yeah. Let's see.
I'm not sure.
Oh, it...
Yeah, there she is. Looks like a female to me.
Yeah. No claspers visible along the tail.
Yeah, I got it.
We're here at this corner.
So I guess we are wrong.
Not here.
Well,
no.
maybe.
Yeah. And these little ones are all in the anemones, right? The
Yeah, I would say so.
orange,
Otherwise,
dark
species
red.
of anemones, of
Oh, the
corals.
skate is flat. Can you zoom out? Or to... Ah. Disturbed snap. Her name.
Could we stop for one second?
Yeah.
See if we can get the it's going the other way.
Yeah. Okay.
Yeah, that's fine. Thank you.
Let's see if we also see some eggs or so.
Yeah. We haven't seen any so far. Yeah.
No.
Okay, keep moving.
Okay.
here. Somebody
Yeah.
wrote his name like an A to the right, T and A. Ta.
Who was here? But yeah, it's
Signature.
interesting. Why are the marks here?
An alien.
Oh, alien. Yeah. Let's come up with some conspiracy theories. They are wondering as well. The deep sea aliens. But yeah, to our knowledge, there hasn't been a research expedition here. Maybe also,
That was maybe the skate, I think.
and it just looked as if it was man-made, human-made.
Don't you think that it was the skate?
The-
Skate marks?
Could be, yeah.
Yeah, laying there and chilling a little bit,
Yeah, and then
moving,
doing it.
shit in the right
Yeah.
posture. More marks.
We have a lot of the little. Are those also the anemones? The little dots we can see, or are those sponges?
Those are sponges, I think. Well, probably a few things over there, but
Yeah.
that looks more like a sponge to me.
Yeah, yeah.
I don't see tentacles or.
Sleepy anemones.
It's like
Yeah. Like a path.
A little path.
Yeah. We're on a hike. Where do we go? Left or right? Or we just stray from the path? Yeah.
You want to follow the path?
No, just into the unknown. We follow our transect into the unknown.
Sorry?
Shall we follow this mark?
There are a lot of marks. Do you think this bottom is our foraminifera? It looks from the structure a bit like something.
We've been sieving the samples this morning, and there was these gravels with a little bit of mud.
Yeah, that's true.
Yeah. Yes.
There are a lot of little stones.
Yeah.
Let's reach our beat. Can we increase the speed to 0.3, please? Thank you.
Polymastia there to the left, and this is a Cladorrhiza, the little tree. That's Cladorrhiza. See now Polymastia.
Delays, you think? Or it's too difficult to tell on the species level from this distance?
It looks like the air, but it's not that clear.
Do you think we should take a sediment sample soon here in this area?
Let's do it here.
Yeah.
Excuse me. Okay, to stop here, somewhere around to do the mark.
Yeah.
Okay.
Thank you.
Maybe here in the corner or so?
Is there a specific spot?
No. Just to get the sediments, not the rocks.
Yeah.
So maybe a little bit more port side.
Yeah. Could do that.
A little bit. Ambition.
Yeah,
About
un
to say.
poco.
Ambition.
Un poco.
Yeah.
Holy shrimp. Oh, do I see a lizard and oryx in the distance?
Yes, we will cover now. Thank you, Francis. That's what.
Do you want to start? We have 15 meters.
Let's cover the mark first, right?
Yeah.
Here, or
Yeah.
where we-
Reach RV, can we stop the movement for now, please?
Here in the brighter part, maybe it's good.
Yeah.
Thank you.
We will have some.
Well, I'm not
Some
sure what.
pebbles.
I'm not sure the one. Maybe this looks like foraminifera actually.
Yeah, yeah.
So maybe
And I don't think they just want pure sediment.
They probably want a mix of everything, right? So they're looking for things inside that more diverse. No, I don't think so.
I thought they just want an area where it's not covered
Okay.
by anything.
Then yeah.
With they, we are talking about
Done? Which of the coffee mods is done?
the right one is done, right? So the left one is, I think, still free.
From left on
I'll
forward.
check. Yeah. Mhm.
If you can zoom in a little bit, just follow the jaws. Which one are you going for?
So we have a team of scientists that are
So you want me to
interested
zoom
in
here? Or-
Yeah.
in
Keep that
different
on the
faunal
jaws.
communities here, and
The boats.
we are collecting sediment
That way.
samples
So you can see it
that we
moving.
will sieve afterwards to see what's living sediment and how it is composed.
Not?
And we already, or the other shift, Anna Helena and Sergei, they already collected another mug of sediment, as you might have seen, and we're gonna do the second one here. There's also a snail just above, but yeah, you zoom in on the important part right now to give also the pilot a little bit of support for knowing where the arm is at the moment and how it can grab the mug.
Oh yeah.
Cool.
Do you have a specific area? Or
Where it's more clean. For instance, I can show you. Yeah. You see?
Okay, that's fine.
If it's okay, yeah.
Oh, yeah.
Yeah.
Then we find other things.
That's where you are. That's the white area in the red. Swing to your right. So compact.
Yeah?
Right.
Up the trains in at the moment. That's why you can't see it. Okay. All yours.
Perfect.
Beautiful, thank you. How do we want to go about with the Lysodendorix?
Oh yeah, the design I forgot
How
about.
about mesmerized by this sampling skills?
There is a sponge we would like to collect.
We have another sample we would like to take, and the distance in the top left corner, there's on the rock a little Lysodendorix, or even two sponges. And the sponge experts are doing connectivity studies on them, so we collect multiple ones of them. If you've been following our dive streams for some time, you've probably seen us collecting quite a lot of them yet, and know everything about our objective here. But in case you just tuned in, this is a really cool sponge called Lysodendorix complicata, and it grows basically everywhere. So we have it here in the different depths that we were, from like 600 meters until almost 3,000 meters. It was just always there.
Just one shade that is actually yeah. It is. It's growing on Angelia parva, and it has an amphipod. If we
It's
could
a
collect
big
the
amphipod.
amphipod, so that would be slurp. Yeah. So here the problem is that this is attached to this.
Yes.
So everything will come.
We won't,
That
like
is
the
not-
Lysodendorix might come
It's
loose,
too big for the slurp gun, maybe.
but you
Can
don't
we
think
pick
we
it
can
with
slurp it?
the scatter minute and suck it then?
Yes. That would be perfect.
But the amphipod might escape.
You don't want the
The amphipod will escape, so
What's going on?
I think the T4
Polynight, right?
because
I mean, we can slurp the amphipod first, and then
It looks like an interesting amphipod to me, but who am I to tell?
So shall we slurp it first?
what's
Francie?
the one you want to start first?
The little amphipod
The little
The red
trap.
one?
Yeah,
Orangey
amphipod.
thing?
Yeah,
Yeah.
the orangey one.
on chamber 5.
Is that an amphipod or a polygid?
I don't know, sorry.
That's a polygid.
Whatever.
Definitely. That's a scale worm.
So we don't want that.
Oh yeah.
Oh yeah?
No.
Chamber 5.
But it,
Are you sure about that, Sergi?
Ah,
Yeah,
yeah, I'm
it's
pretty
look-
sure. I went with the polynights, yeah.
It has legs, huh?
Yeah?
Yeah. I don't think it is,
That's a big one.
but it has.
Segmented, right?
yeah, it has
You see
segments.
segments.
Yeah,
And you see like a kind of a...
Take a look.
Yeah.
and
Yeah,
big
go on,
eyes and some mouthpieces.
But what is weird is the legs.
Yeah,
See?
small
Legs.
Ah, no.
cockroaches.
Yeah,
See you
right.
later.
You're slurping it.
Who's going first?
Yeah.
Are we going first?
Yeah, I thought it was like a
We're going to
polynight
slurp
was
first.
coming here like crazy.
But you don't think that if we slurp this would come off? The Geodia?
The Geolia will come.
No, I mean the Lysodendorix will come off,
I don't know.
because that way we had.
Yeah, you should slurp it.
The thing is that it's attached to the Geolias.
Yeah.
And they will be
If you
messy.
take a lever,
Well, that's on them.
Yeah.
I mean, they will start by slurping the amphipod or
The amphipod,
whatever.
yeah.
Maybe piece of
There's plenty of
Uh,
amphipod here, right?
No.
plenty of everything.
So,
Yeah.
I explained the situation.
Okay.
We can slurp the amphipod. If we can slurp the sponge without taking the other two large sponges, that's okay.
Yeah.
This sponge is a little bit brittle, so it may break if you slurp from
Yeah,
certain distance.
Yeah.
If you get too close, the other ones will come.
Yeah.
So maybe we can try to slurp.
So you
from-
want us to make it impossible
Make it possible.
Yeah.
Make the
Mission
impossible
impossible.
possible.
Yeah, make it.
Make the impossible.
Okay, so what I'm
possible.
thinking of is bringing the starboard arm around the white sponge, and we slurp inside the jaws, so we hold the sponge down.
That's perfect. That makes sense.
Isn't that something so easy? Okay, let's try and make this happen.
We have an amazing ROP team here, making the impossible possible and always coming up with amazing solutions
Yeah, but I
for
come around and I hold
the
right,
different
so I'll follow you around
samples
first.
we would like to take. So yeah, we're really blessed here on our expedition with this team of ROV specialists.
Okay, se sabe.
Porque es que ya es que voy con
Sí. Bueno, no sé. Hay que.
y que él intenta tocar esto. Vamos a ver
¿Cómo es? Lo hemos visto, sí. Sí, sí. No, okay. That simplifies everything.
Staying there.
Is it a
This is my house.
Zoom in for me.
not so much?
Sorry.
This, yeah. Is it like a ping pong carnivorous sponge?
It's kind of a sponge.
And the ping pong balls? There are these ping pong carnivorous tree sponges.
No, it's not.
That's
no,
not them. No, they look different.
Varia,
Yeah.
no. Those are big.
Yeah. I didn't see them yet.
I wanna pay attention to this because it's really. So they want the orange tips that way.
Yeah.
Okay. So that's the reason to keep.
Yeah.
I'm frozen there, so I'll take a look at
Yeah, because you want to be preserved in
it.
the
You might get a little bit of the darker brown sponge.
It's okay. No problem. Shall I come with Andrea?
No, you can leave it there. That's fine. Thank you.
Sorry? Yeah.
That's fine.
Come on, listen and
Okay, let's go back and touch turn off first.
Yeah, that's not.
You can keep it for that.
Yeah.
Perfect.
Oh, nice.
It's okay.
Stop.
It didn't go through. Second, it didn't go through Sterling's and the nozzle.
Stop and just
Yeah.
time.
Just look at the nozzle and fall out, check it out.
Come out, little Geodia.
There you go.
So we're trying to sample just what's necessary, but it's sometimes not so easy to separate everything from each other. So yeah, we're trying to get at least the Geodia out again, if it's still stuck in the slurp house.
Can that sponge go into another box?
Yeah. No problem.
So we can do that, Rob, and then just suck the whole lot into another box if it's not coming.
Yeah.
So that probably gets stuck.
No, that'll go through.
Okay.
I'm not going to move the box. What number would we put that other sponge into?
You want to have a
Two
look?
biobots.
Oh, there's the one.
So it has to be.
The number we have.
The number 5 already has one more, please.
This is slurp, and here is what is the
So, slurp or biobots?
Slurp.
Slurp 4.
4.
Yeah.
Alright.
And then we have the Geodia and
Okay.
the Solenogaster all in that one.
There was no Solenogaster on 4. Now there will be a.
Okay, it's in 4.
Perfect. Thank you. Now, thank you.
Hi guys.
There was no Solenogaster.
No.
Huh? Now there is a two Geodias and one Solenogaster.
And nothing we've got in number 5.
I'm not sure. Maybe the first Geodia.
Yeah.
They did
Let's
one
check.
big in the 5.
But we will know.
Yeah, but
We'll
you
see.
have another
But
one
let's
here.
check now
Already on the phone.
if they know. Did anything go in slurp 5?
Yeah, we put the white bit into slurp 5.
And the first other
one.
one.
Yeah.
Yeah. Good. Thanks.
Okay. So I'm good. Dos.
Oh, that was 13. Number 13.
No, ya me traen. Ya me
I have too many papers here.
From my side, I'm okay. Anytime you.
Alright, dude.
Where you go?
What?
No, fancy. Where you go?
Yes.
Yeah, shall I help you? Something.
Yes, it is.
Sorry.
I have it
That
completely
was-
under control here. No, there was just the papers that were in other orders. But yeah, so we have first part of this cast on here and the
Geodia, and then another Geodia with the fan-shaped sponge pieces.
Yeah.
That's it. So sponge soup, two scoops there, and those are free.
You want
Yep.
any product?
No.
Table space.
Yes. Yeah. The shape looks different. No?
Bridge RV.
RV bridge.
Can we resume the movement at 0.2, please?
005 and 0.2.
Thank you.
Okay, we're continuing our exploration here. And after we've climbed a very steep wall slope of the seamount, we've arrived at a more flat area, more sedimented area, and it seems to be covered with foraminifera, at least at some parts.
Yes. Yes.
Sorry, you wanted to say something?
No, just that I think that they are looking for those orange tubes. I'm not sure what it is, but
Which orange tubes?
You remember there were a couple of orange tubes before that we were wondering
I
about.
thought like those were broken off pieces of the asbestopluma sponge, of the carnivorous sponge, because at its stem it also had more orange shade.
Some are, yes, I think, but not all of them.
Yeah. In the distance there's a fan-shaped one, right?
Yes.
Yeah.
Well,
A fan-shaped sponge. Yeah. And a carny.
yeah, I was top blue mark.
Still a lot of shrimp hanging around.
See if
On
I
the
can
bottom.
I can get this one. There.
Ooh, no, I didn't want to go in the plume.
Very white. So I'm not sure what it is.
What is?
The sponge, but I guess that is the same that we
Hmm.
colored.
Do you already have ideas how you would name the seamount?
Uh-huh. Are we talking about that?
Because it still has no name, it's a blank one.
Seamount, the anemone seamount.
Yeah, I was also thinking of something like that. Because we saw so many nice anemones before. Is there a fish?
It's a skate.
Is it? No.
It's a fish. It's a fish. It's a fish.
I'm not touch it.
Yeah, eel pout maybe?
Not sure. Big one.
Hmm.
So it seems that the summit is more sedimentary.
Yeah.
I think it was the same this morning, right? A very nice slope and then
Yeah.
more muddy gravel on the summit.
Yeah, and in comparison to the earlier dive that we had at this mount, we are now at in a more southern region, and the dive
southern?
before, we are more in the south now compared to the dive we've had before.
And the depth?
In comparison to the earlier one,
Yeah.
so the earlier ones were between 1.2 and 1.5,000 meters depth. And this one is now 1.4 to 1.6, so a little bit deeper.
A little bit deeper.
Yeah. Further south and a little bit
It's a
deeper.
crinoid there. I think it was a crinoid covered
Oh.
with anemones.
Oh, and have we talked yet about that we are now on the eastern, that this seamount is on the eastern side in comparison to the western sides that we had in our previous dive, so we are also interested in comparing the seamounts east of the ridge to the ones on the western side, and also taking then the flows of currents into account and what they might do the community structure.
And the thing is that historically the western part has been more well studied by the Norwegians, while not much has been done in the eastern part. Probably nothing, maybe. But I don't want to say that because I don't know. But in terms of publications, you have some nice studies on the shoes. I think that there are some preliminary studies on the voids, very preliminary, and on the Mons treasure as well, but not here. When we collect the biggest sponge, that was...
We're going...
it's like we
we were already like 10 meters shallower, but it didn't differ much. Let's check the depth profile. Yeah, there's a part I think where maybe at it now where it goes, the slope is slightly going a bit down, maybe 20 meters in total or so, and then it goes up again on a little peak, and then it's
that
fairly flat again.
That peak looks nice.
We're looking forward to the peak.
Maybe where the direction changes.
Yep.
Do you know how much is the resolution that we have in this area?
30 something, so fairly good.
35.
Yeah, 35 somehow, more or less. Yeah, so while we were planning these dives, we had different different resolutions of the seabed already. What's going on?
Okay.
Oh no, I think our pilot bumped her leg or has problems with the chair. Happens to the best. Yeah, so we had a different resolution, so the accurateness of the seafloor data that we had before was different to each other depending on the sites that we visited. Sometimes was more clear and sometimes more coarse.
And then it will be turned into port.
And in the back we have one of these bigger fish again.
Still going up.
Similar to the one we've seen before. Where's the fish sheet? The fish sheet is not there. But it looks like a big eel pout actually. But it just has a very big back.
They're turning up.
There are more of them. Oh, this one has a big mouth. like it's like these big, the fish, demersal fish sitting on the ground and just having a big mouth that they can snap open and close again.
How big is that fish? I think.
Huge. Yeah. Something like that. Oh,
There's plenty of them, no?
yeah. Oh, is it the one that just swam off?
Maybe. Yeah. So we're trying to get the proper direction because we are at the very top.
Ah, yeah.
Bridge RB.
Okay, great.
Can we change the bearing to 350, please?
50.
Thank you.
These are Sediment.
Mm-hmm. Some Ascidian, Carnivorous sponges, and Geodia, and soft coral on top as well.
Yes. And some polymastia.
Mm-hmm.
That's good.
More fish. And are there some of the fan shape in between, or are they all? They are all Rosalida, no?
But
Mostly, at least.
so that one here. This one. You see? Could we stop with well, maybe too late. That looks like Geographic to me. You remember the
Yeah,
Geographic that was so
I
common?
actually remember. Yeah,
So
that's been a while.
Do you want to look at it?
Yeah, just look at it, not collecting it. It will be enough. Because I actually think that Paco collected that one in the
night.
in the night, during the night.
And there's a sea spider as well.
Sit down so you can
The beautiful red, yellow one. Yeah, we came to a spontaneous halt. So now we have a little bit of a sediment stir.
Can we stop the movement for now?
All stop.
It's not really washing. Is it?
That's just in front of me. I'm watching this camera, so I'm not moving.
But I mean, it's not really washing the clouds. So you think
It's coming.
Ah, no.
Have fun.
So I am too nervous.
It's fine.
Yeah.
Now the view is clearing.
So here you have the fish, the
Yeah.
sponge.
And
Okay, I
you-
can start with the.
Do you still need the lasers? Otherwise it would
Let's
be
see how
nice
big is
to
that
zoom
fish.
in on this
Lasers
one.
off?
Yeah, lasers off.
Thanks. It's a nice little community there. Oh yeah.
You hold that for a while?
Hold for a while.
pan slightly to the right and zoom in a little bit.
We're getting some instructions also from our visual and communication colleagues who can see the screen as well up in the dry lab in our office area. How still everyone is just in this picture, like they're staring for a painting.
That's a very nice image, thank you.
Thank you.
It is a very nice image.
Let's see that. I think over there. Two Pokemons.
Yeah. And
The little polymastia. Yeah.
little cute little polymastia sponge in the left corner, and the buff is awesome. Yeah, so they are all just hanging out together. Oh, okay. Now the fish has enough of posing, or not.
I was just getting comfort.
Are those little parasites, you think?
Maybe, yes.
Yeah, on its tail. And the shrimp also wants to be in the picture. And little...
Hold the camera still if stuff are moving in normally. So if there's a shrimp passing by, don't try to follow it.
Not following the shrimp. Going to the parasites.
That's parasites?
Yeah, I think so.
Eating on the fish. Can you zoom or tilt a little bit so that we can see the head as well? Yeah. What are you thinking? I think we
Very
can
good.
leave it in peace, yeah. Yeah.
Moving on.
Hold on, let me check the sponge.
We distracted Julio from the sponge. Yeah, I forgot about them. The potential Calucothias, the one that are more brownish, but also fan-shaped around the Rosalidae sponge. And what is this one?
That's not sponge.
No, is it the bryozoan? Yeah. So we have a big Geodia sponge, first of all, on which then several other sponges, also carnivorous sponges, grow upon, and yeah, these fan-shaped ones.
Okay, I think that we are good to go.
Cool.
Right?
Yeah.
That's it, we go?
Yeah, we can turn the lasers back off, on. Thanks, and continue. And some Rosalida in between. And what's this association here?
That's a rock.
Only? But there's
Rock
some...
with the geodia on top.
And some of the worms, Cephalidae, Carnivorous. And this one is a dead sponge on the side of it. Or front of the Calucothia.
It's a bit hard to say.
Yeah.
It looks like a Calucothia. Several going together at least. Okay.
I thought I saw ice, but it was just some dark pebbles. Something staring at me out of the dark.
Black coming after you.
I imagined a fish lying down there, but no. I think those were also the anemones the small little dark orangey ones that we see sometimes, often in between here.
Maybe the alien
Yeah.
following us.
Oh.
How you call because an UFO is when it's flying, but when it's under the water, it has a name.
Does it?
USO, no.
Unidentified swimming object?
Yeah, I guess.
The USO. Yeah,
USO.
seems legit. Is Holly a user? No, it's
because identified.
It's identified, yeah. Another of these cool fan-shaped sponges.
It happened to me today when I tilt the camera to the left, that it goes like that.
Yes. If you zoomed in too much, it will.
It will.
Yeah. The idea is, if you want to zoom in on that, centre up in the camera
And then zoom
and
in.
then zoom in. Because the camera...
You see, but now it's not zoom in and it's still doing it. Yeah, it's a little bit different than the other days. Yeah.
Yeah.
Has been happening also before.
Oui.
Feels like there is something physical avoiding it. Well, could be.
In the steering of the camera or in the zoom?
This one.
In the steering. Yeah. So Julio, you can
Jellyfish.
also probably see it, has his joysticks there for an... Oh, okay. Oh, that's... Oh, like very delicate bubbles. What do you call it? The soap bubbles?
Soap bubbles.
What you do as a child, or maybe when you're older as well, you have this soapy liquid and then you blow bubbles.
Yeah.
Soap bubbles? Yeah, it looked a little bit like that anyway, and I will look it now.
Can we start movement 340 bearing at 0.1?
Who knows? Asconema? No.
Thank you.
I locked the wrong one.
Rosalide. I would go for Rosalide
Oh, yeah,
because of
it
the
just-
size
yeah,
and
true.
it feels more like
Yeah.
it.
I agree.
It's a fish over there,
Where?
right in the middle.
Ah, the whitish one. Yeah.
No, to the left. Well,
No?
this one.
Yeah, I mean, yeah.
Yeah, well, yeah.
Oh, I'm getting tired. We started our shift at
You see?
2 in the morning.
Now it's-
So
now it's
and now
out.
here it is almost
Now it's
2
out.
again.
Yeah.
If I go to that, well,
Yeah,
now it
and I
works.
can feel it.
Now it works.
Is that a shrimp behind it?
Mhm.
Yeah. Not a small fishy. Oh, much more. Yeah, or at least another of the E-pouts coming up. Is that a carnivorous sponge?
Yes.
And yeah,
That looks
like a spider?
A spider maybe, or shrimp, I'm not sure. Like a dolphin, you see on the rock?
Yeah. Mm-hmm.
Maybe it's not.
But it's like,
The good thing is that I have a sample from the night dive.
Mhm. So we can check up on it.
The night or the yesterday.
Yeah, time is very fluid here. Time and space. Oh, another one of the fish. Let's check this. I think do you think we're at this corner now? Yeah, it's now getting more square. Yeah.
So it's a little bit more than well, it's more
Maybe
than a half.
here.
Yeah.
Yeah. So now comes a quite a flat area. Maybe we want to speed up a little bit.
Sure.
Yeah.
Can we speed up a little bit?
Yeah. Just tell me if it's too fast.
Yeah.
I don't think you will because it's very homogeneous. Small one.
Yeah, and also curling up, like the ones that the other shift saw on the night dive.
Those were small, right?
Maybe, yeah. Oh. They really like to hang out here on this one. So it seems to be, seems to be an important area for the fish here.
Can we change the bearing to 330, please?
Many shrimp also hanging out here on the ground.
Thank you.
It's full of fish, right?
Yeah, yeah. More coming up. Oh, there's even 3 of them. The longer you look.
4.
Oh, yeah.
Probably more.
In the distance also. Yeah. I first thought, I think it's an anemone probably on it, and that makes it just look like a hole. Yeah.
If I turn my head, turn the head to port, you can then center your camera up and that'll get rid of the shadows and the eye. Will that be okay?
You will tilt port.
Yeah. If I turn to port,
Yeah.
right, now you square up the camera. Like that.
That's not enough.
Maybe even a little bit more. Yeah.
Of the fish. Yeah. Where do you see the second one? Oh yeah, that's there in the distance, yeah.
They want us to double check what's on the bio boxes for the next switch
Oh yeah.
to tell them.
Yeah. We have a lot of space in the starboard side. 3 are still free, and I think that's it. And more Lysodondoics would be good, I guess.
Yeah. Yeah.
We don't have Lysodondoics in the last, and just in the 4th one yet.
Well, but I'm all worried about them if they want some crustacean. So they cannot put it on those because it's already amphipods in here, amphipods in here.
Yeah.
Maybe this one they can because it is on Doris.
Reader, can we increase the speed to 0.2, please?
And then here, fancy.
Look, there's
Thank you.
the interesting bunches.
Is that too fast?
No, it's good. Thanks.
This is empty, right?
Yeah.
And
That one.
we'll put something here. Correct me if I'm wrong. No? Maybe not.
I think that was in the previous dive.
Okay.
I can check. Was big bio box 3. No, we didn't note anything down for bio box 3. But I will make it more clear here. Do you want to look further?
Well, just tell tell them that
More fish and
that's
facility.
it. If you agree with me,
Yeah.
these three are empty,
Yeah.
and this is empty, right?
Yeah.
And here it's only the max.
Mhm.
And this is what's in here. Cast 10. Cast 10. But yeah, that makes sense. Geographic or rock? No, I think it's rock. Actually, sorry. Big sponge there.
No, in the distance there. Oh, and then another eel pout visits us.
You see, that kind of moves to the left. It goes like
The
the camera.
yeah. Mm-hmm.
It's just
Reach ROV. Can we change the bearing to 320, please?
you have
Thank you.
these two
The
worms.
crusts. Ah, the worms, yeah.
Now this is the anemone.
And a lot of holes in this one.
Now big rock. Well, big rock. A rock.
Ah. Is that our little pig? We're now at 1,447 meters.
So that's on the peak.
Yeah.
It's coming up rather steep now.
Mm-hmm.
Will be very steep.
That should be just very shortly. Yeah, that should already be the top. I think. Can we slow down a bit while we climb the slope? Thanks.
Big carnivorous sponge.
A lot of them.
I say what is that?
Looks funny. The big fan sponge on top now. Is it fallen over?
It's the same one.
Yeah, it's just the direction of it seemed a bit weird. And there's a nice overhang of anemones and lots of carnivorous sponges and the tube worms. And of course the sea star is there as well.
Yes. Stuff or what?
Yeah.
Remove the lasers, please.
Yeah.
Do you think it's finding food in there, or just cozying up in the
Sure.
nice soft sponge?
Yeah, sure.
Or hiding from the squid that we saw before that was hunting fish here? Now we also see the many little pretty anemones around. And the soft coral, little soft coral underneath. Yeah. But this sponge has really built a nice,
Nice house.
yeah, nice little cave almost. Yep. I just now see also that there are sea stars around on the carnivorous sponges, huh?
Yeah.
So are sea stars somehow like they eat sponges, and do they also eat carnivorous sponges, or are they like don't like the sticky texture of them? Is it somehow it doesn't seem to bother them,
I don't know.
or maybe they don't know yet
I
that
don't
they
know.
shouldn't eat this?
I wouldn't go for the carnivorous sponge. I would go for the fluffy one.
Yeah.
But no, I don't know. Good question.
Maybe the carnivorous sponge is eating the sea star here. That would be a game changer. But no.
Shall we keep moving?
Yes,
or-
let's keep moving and leave them in peace.
Yeah. That's the next one.
Thanks for stopping. We can
Okay.
continue, yeah.
Continue. Yeah.
Hello. Hello, Paco.
Hello everyone.
Paco and Ananina arrived. The next shift is taking over.
The big one.
The
Sorry?
2 o'clock in
No,
the
I
afternoon
just
till
arrived.
2 o'clock in the
Big
morning
one?
shift.
This size.
Oh,
It's only
okay.
happened
Same
once.
species we saw last night?
Probably, yeah. And then the thing that you collect, I think it's Geological, the fine shape, tiny
Okay.
one. I think
Okay.
it keeps happening all the time. And
You
those,
mean the first one?
the one that is thin,
Yeah. That was a bit transparent, like.
exactly.
But it looks more massive than the fan shape we collected before. Wow. Okay.
We should
So
be
we're not
here roughly, so
Okay.
about halfway through.
Okay.
Yeah.
Plenty of amphipods.
I think we're reaching this corner now.
Yeah.
So,
And many fish all
you
around.
have very good for samples. This is a little bit crowded, but you have all these three
Yeah.
and this one. And here the two marks are
Are done.
You can put things on it if you want, or if they want. But
What
you
about
just-
the slurps?
So the slurps, Asconema with many amphipods, and there is Solendorids, amphipod,
Yes, please. Thanks for thinking of it.
Solendorids and the crust, pieces of one of the big ones that we will take in very nicely. A Geological that came by accident, and here also a Geological that came by accident, and the Solendorids. No amphipods
Okay.
Yeah.
that I saw. There may be.
But which one? Is there any slope that needs to be is precious, or when we can put something on? Can we put anything on top of like the 4 or the 5, the 3 or the 5?
Just don't put the Solendorids on any, because there is the Solendorids.
Oh,
everywhere.
there's a
Sure.
nice
That's
fan-shaped
fine. Other than
sponge
that,
coming up.
You can put things in any.
Okay.
But
As
the
long
thing
as
is,
you are aware that there are a lot of amphipods in
exactly,
those two.
we don't want to mix amphipods from different stations.
Yeah, but
Other than that, for me, it's just not mixing the Solendorids with the Solendorids, and because there are the Solendorids everywhere.
Yeah.
Always
Can you
Solendorids.
stop the
You
movie
don't need,
for now?
but
Sure.
then here you have plenty of space in the bio boxes. Okay. All this is free, and this is free.
Perfect.
Okay.
Thank you very much.
Very welcome.
All
See you
right.
later. Bye.
We're handing over to Paco and Annalena. Enjoy the rest of the dive.
Okay.
Bye bye.
Okay.
Okay.
Thank you.
See you.
Hey friends, all the best. See you later.
Hello everyone. My name is Paco. I'm curator at the Museum of Evolution at Uppsala University in Uppsala in Sweden. And
I'm Anna, and I'm from Hamburg, from the Senckenberg Institute. It's our pleasure to go to the next dive.
yes,
Fly along
yes,
at our unnamed seamount.
we were there a little bit last night. Then we had a good night's sleep, and we're taking over this second dive to the unnamed seamount at 76 degrees north on the eastern side of the Mid-Atlantic Ridge, the Arctic Mid-Atlantic Ridge. We've already got some samples in, but we still have quite a long way to go for this transect. So another couple of hours with us, and some more sampling to explore this never explored before seamount. So we are the first ones, the first scientists and the first humans actually to see a little bit what type of animals and habitats. And so far we've seen already quite a lot.
Yeah. But the real beauty is really shown in the lab afterwards when we look through all these samples under the microscope and sorting the sediment. There are lots of critters living in there.
Yeah, because we had a first dive already, so some of those samples were already processed and they look promising. This is the second dive we're doing here, and then we might have a third one, so that at least we have sampled several places. We will see.
The samples are so super precious to us because they're on the eastern side from the Atlantic Ridge.
Yes, yes. We know far less about the eastern side of the Mid-Atlantic Ridge than the western side. Maybe even we know nothing about the eastern side. I don't dare to say it, but it's a little bit like that.
At least there's nothing published in obvious sources.
Mm-hmm.
So we were unable to find anything.
Yeah, yeah.
That's 005.
We seem to be on the kind of a ridge.
Yeah, it's going down, Paco.
Okay.
Can we start the movement of the ship bearing
000 north at 0.1 please.
Yeah, 000 at 0.1.
It's still not zooming again.
Yeah, we would, if not, we want to have it come to the northwest.
So we don't like it when it goes down because it's not easy for the ROV pilots to swim, to fly,
And we see less.
and we see less things. Yeah,
So,
it's not
it's a-
it's not good for scientists as well because the way the camera is positioned, it's difficult to know.
Well, and there's a
What you
slight
see.
upwelling, so we have a lot less visibility to the water.
Yeah. Yeah, a lot of
Okay.
action in the
Mhm.
water, a lot of crustaceans.
Yep. And some sponges.
And some sponges at the bottom. Not a whole lot. Some pilolavas. We hadn't seen pilolavas for a while. So pilolavas are those round rocks you see at the bottom, and this is when magma comes up at the surface, meets water, and the magma solidifies very rapidly. So the minerals in the magma don't have really time to grow. This makes a very fine-grained rock that we call basalt. So whenever you find pilolavas, you know this is really the bottom of the oceanic crust. This is the top surface of the oceanic crust. And when you find it on land, it's an indication that there was a sea before here.
Where do you find it, for example, on land?
So on land, we find it in a lot of actually mountains,
Can we increase the speed to
Like the
0.2,
Alps?
please?
like the Alps, for instance.
Thank you.
Change of tape in 10 minutes.
Okay.
I used to be a high school teacher back in the days, and I would take my students to the French Alps, and especially there's a place called Le Chenaillet. We would walk to the top of that place, and we would find pilolavas exactly as you see them here, except they were on land, they were in the Alps, and this is just because the Alps is actually a confrontation of two plates. Before there was an ocean in between, and it was crushed and taken up.
And here the ocean plates are spreading apart.
Exactly, here it's
So very,
the opposite
very
movement.
slowly.
Yeah. We are on a mountain that is right next actually to the mid-Atlantic ridge, so this is probably the youngest mountain that was created on this part, on the Eurasian tectonic plate. And as we get far and farther away from the mid-Atlantic ridge, from the middle, we get mountains that were created that are older, that were created a longer time ago.
So one of the questions that we're looking into is if this mountain ridge acts as a barrier to see how things from the east and western side are related.
Exactly.
For this we take samples and fix them appropriately and look at the DNA from all kinds of specimens.
Yes.
Some of them have larvae and some of them don't. So we're looking into how this influences their distributional patterns.
Exactly. One of the long-standing questions in marine biogeography is are mid-Atlantic ridges natural barriers for populations, for species? But for that, to answer that, you need to be able to sample on either side of that natural barrier of the mid-Atlantic ridge, and
fix them straight away appropriately.
yes, to try to get genetic data, and we're the first ones to actually be able to sample on the eastern side. So this feels like a privilege to us.
Yeah, it is a privilege.
And this afternoon, Rob is our ROV pilot. Andrea is our co-pilot today. And
Paddy?
Patty behind is our advisor, supervisor. You know, I was with a school this morning talking with little kids, 8 years old. I had so many questions on the ROV. What is it made of? How can it withstand the pressure? How comes there's electricity in the ROV if it's in the water? I wish Patty would have been there for some of these questions. I was like,
be able to answer some of those. Yeah, good questions.
yeah.
If you have them, Paco, let me know and I can give you the answers for if there's any more classes.
Yeah, absolutely. Thank you. Yes.
Several of the fish.
Yeah. So that's something we noticed actually during our first dive here last night. There were quite a lot of fish on the seamount.
Contrasting the seamounts that we'd seen before, where we mainly saw eggs
Yeah.
from skates.
From skates. Here we saw one skate only.
Yeah.
We will need to check the footage during the night, but it doesn't seem like there's a lot of skates or even eggs. We haven't seen eggs at all.
Would it be possible to stop, please?
Yes.
So we are at 1,455 meters depth, and we're gonna take a little stop. It would be an opportunity to study the ground, but also to change the tape in 5 minutes, because we are recording this, of course, and our tapes are 2 hours long. We need to regularly change the tape, and we make multiple copies of these tapes in case something goes wrong. It's always better to have copies of everything in science.
Tape stopped?
Oh, sorry. No, yeah.
Okay.
What stopped?
What are these potato-shaped things? Okay.
Sponges.
Yeah.
I'm just laughing because sponges are everywhere. Even if you only see rocks, then high chances you'll see some sponges as well. Anemones.
Do you think we could slurp it? Because we've been seeing this everywhere. I think I know the species, but for this I need confirmation from. I need to connect what I see with the sponge as we get on board.
Yeah, if it's not too far away, yeah, we can scooch forward and get it, yeah.
Yeah?
Which one's that?
No, but I think they're also in the front.
Or some are
Hmm?
closer. Yeah, maybe
Which one?
if you zoom on those, maybe they're the same. I'm not
Yeah,
sure.
That's what. Look at these. Let's look here.
At least they would be easier than to get than these ones, which.
Let's look at this one
Yeah.
first.
But
I don't think so. I think any of those, yeah?
yes, but this will be easier to get than this.
Yes, but isn't it easier to take in a shovel?
This will be easier to get in a
Because
shovel.
you can get the little rock what it's sitting on.
Exactly. Or are you thinking maybe the slurp would be easier for these little rocks?
No rocks for the slurping.
No, not for the shovel or slurp.
How big is that rock?
These
Look
little
at
pebbles.
These ones.
Oh yeah, we get those.
Or this one.
Yeah.
This one.
Yeah, we get that one on the right there with the scoop.
Yeah? Okay.
Yeah. You want to do that too, or like
But can we do a zoom on one of them to have a really good...
For sure. Which Which one is easiest for you to get?
The one to
Like
the
this
right,
one?
yeah, that one.
This
Yeah.
one? Okay.
Okay.
Yeah.
Now lasers off.
Yes, I'm gonna wait for.
The rough is moving. So
Yeah,
we
yeah. I'm
can't
gonna
do
wait
it.
for the rough to settle firmly on the ground. And anyway, I think we should maybe wait for the tape change.
Okay.
Like this will be no stress, and
That worked.
and like this we can do our little Zoom session. Is the RAV on the sitting?
Yeah.
Yes. Can we maybe zoom on this? Yeah, on this one. We're gonna zoom, and then there will be a tape change in 2 minutes.
Which tank?
No.
With a shovel.
But it's in the bio box.
So if they're picking it up with a shovel,
Don't you want to go on this one so we have that one clean?
3?
Yeah.
Yeah. So this is starboard, right? And this is, I never know the name of it.
I'm also confused.
On the left, what do you call the boxes?
Bio.
Bio boxes.
Yes, but
Oh, outer.
outer board or
Outer.
outer?
Yeah.
Okay. When we take the shovel, can we try and get these crawling units in? Just wait for a second. It will walk towards the sponge.
Will they not fall through the holes and scoop?
Well, we could try.
Yeah.
Tape change in 1 minute. So whenever you're ready, Andrea, we can do it now.
1, 2, 3. Stop.
Stop recording.
Well, this crawling unit there is an amphipod.
Mm.
Yes. You ready?
As soon as the new tape is on.
So while this one amphipod has crawled out of the picture, the one I'm actually looking at is one walking towards the sponge.
Recording.
And now behind, no, look, now it's going directly to the sponge that you want to sound.
Oh.
So.
Perfect.
Well. Okay.
Okay. So this will be cost. Yeah, I'm good for the image. Oh.
Okay.
Ooh. But maybe we can see it.
Oh, that's tricky now to see on this camera what we're doing.
How is it, Rob? Can you actually see what
see?
you will be doing with the scoop?
Yeah. Move the drawer back in.
Okay.
Okay.
Yeah.
So I'm writing the date, coordinates.
Can we
Is
stop
it a
the
zoom in the
movement
HD?
for now, please?
Keep going.
Thank you.
We are going to sample the little sponge sitting on the pebble because it's a little bit everywhere. It seems to be quite a common species, but one of the main problems we have with sponges is that they never look the same when we bring them on deck because of pressure, because they're out of the water and so on. So we have a lot of difficulty connecting the species that we collect with what we see on water. And here we're privileged enough to have ROV, high quality, high resolution images that we can actually connect to the sample. So it's fantastic for us to be able to say, okay, this thing, we know what it is. We're collecting with a scoop. Scoop method is very practical.
Where am I on it?
Yes, you're perfectly in front of it.
Yeah, where is it?
Can you show? Yeah.
This one, the little white one. Just if you shove it from a big bag, we'll get some rocks with it. That's fine. There are a lot of little encrusting things on the rock, like small anemones and some polypid worms, so we don't mind when you get a few rocks.
Yeah.
Of course. I will write plus pebbles.
Okay, zoom out.
I'm zoomed out, Max.
Port or
Outer board, yeah. Outer side.
Other side.
And bottom left. Here.
This one.
yeah.
There's nothing inside. We cast 14. It's good to see. The mug hasn't gone there. That's interesting. It means it can stay actually if it's clean.
Has it been used? Both mugs are full. Yep.
Yeah, this one has been used, but not this one. Here, there's nothing. But the mugs have been used. Yeah, they're full.
Yes, but if you want another sponge piece, you can put it on
I
top.
think so because it looks clean.
So if you have a picked one,
Yeah.
that can go on.
Okay, cast 14 is
Still alright.
in. Yep.
Is that everything for here?
Sorry?
Is that everything for sampling here?
I'd love to have one. Is it enough for sampling? Yes. I'd love to have one look.
Yeah, we
Just
can
to the
have one
front.
more look, especially if you see something,
Yeah,
on
but
other pebbles or.
Yes, but I want this to be stopped first. Zooming in, out, anything?
No, I just want to know what you want to do, that's all. Do you want to zoom, do you?
I want to zoom
Yeah,
just to look. Just leave it out in case. Discover treasures. Sorry, my eye is so focused on apricots, I only see them everywhere. Or the only thing that's moving on here. Nee, fly on.
Gives a break.
I mean, I would be interested also to get this little sponge. If you
Can
think
put
there,
it in the same box?
we can absolutely
now
put
that
it.
the shovel is out?
Absolutely.
Okay.
Can you zoom on it
I'll try.
fully? But maybe you're already at maximum.
I'll try to get it set yet. Just this is
Yeah.
sorry, we'll go back to the sponge in a second.
Sure,
I'd
sure.
just like to have a look here.
Do you think it actually sensed that we were opening maybe a place to find food or?
I do not know.
So I hope you can all see on the left this little amphipod, this little crustacean walking on the sand, on the mud.
Well, excuse me.
You can see the bright eye, or I guess they have two eyes.
They have two eyes, but in some group, this is molted together.
Oh.
It's like a cyclope.
Mm.
So it appears to be one eye, but it's actually two.
Very nice. We can do a little bit of behavior observation. It's very rare that we can see those animals in the wild behaving.
I'm still impressed by that, being like 1.5 kilometers below us. That we can look at these little creatures
Yeah,
sitting on this vessel and looking at something that's 2 centimeters in size.
absolutely. The little, so of course I'm not looking at the sponges next to your amphipod, but the little white sponges I think are actually Tentorium. But I think we'll try to collect them as well for confirmation. They're a rather common deep sea sponge in the area, all over the Norwegian and the Greenland Sea.
Okay. A few seconds more. And we will take a shovel again, and we will go into the same box as we just did. But we will take this white sponge that Paco will show you.
Yes. So I think it was, yes, one of these white ones. So it's next. You have the this, and it's right next to it.
Exactly. You
Yeah.
got it. Like that one.
So just leave the lasers there on it, and come down to there now. Just leave it there as it is. You can come down. So it's just to the top of the laser, Andrea.
Yeah.
Yeah, exactly. It's very close to the laser. So we're still using the scoop. The scoop is a really practical tool, especially when it's kind of soft. We don't have to break into a big rock. And then at the same time, we can take some sediments, which will be sorted on board later on. Nothing will be lost. Everything will be studied in the lab and processed.
It's going in, what is it?
Oh, it's a lot.
Sorry?
It's in.
Yeah.
Yeah, it's in.
It's in. You can even move a bit back.
Yeah. Don't take too much because we don't want to
We don't.
Yeah.
cover everything in the bio box. And
It's a little problem now.
yeah, I don't know if you can.
What's that, Andrea?
Can we move back a little bit with the shovel? Thank you. Forward.
Or can you shake a little bit the scoop
Yeah.
to get rid of some of it?
Yeah, which means instead of it blowing,
Towards the other way.
let's go very soon.
Look at this amphipod now. In between its two places, we suddenly
Oh yeah.
things changed.
Oh yeah. I
I
see.
see the shaking.
Yeah. So we're shaking a little bit the scoop to get rid of a couple of
Mud.
stones, some mud. Again, to give you an idea of size, the lasers you see are 10 centimeters apart. 20.
Well, you have conflicting information.
Mm-hmm.
Can we ask the master? 10 or 20? 10. Okay. sorry. No,
No, no.
we were told 20. That's why we kept saying 20. This is yesterday. Go to 10. That makes a lot more sense.
I'm going to turn it towards the camera. And I think it went in the back left, either right hand side.
Oh.
Still too much sediment in their back though, is that?
You think? I mean, the risk now is that we lose the. I think the sponge is inside. I think it.
I think it went back in the left corner.
Yeah, exactly. So it should be okay.
We put this much into the buy box.
It's not
What do you
this
think?
one.
Yeah. Sorry.
Just a second, Johnny. I'm not entirely sure. I thought it went in.
I think it is in.
And it's at the very end of the shovel.
Yeah.
And I think, yeah, just.
Oh, good.
we lost some mud, which is good. The same as before.
Yes.
Port draw on the back.
Yeah.
Taking more sediment?
No. And then we
Our
have
part-
enough.
I
sediment.
think that's fine. It's
Okay.
Because the other parts in it are the coffee mugs, and they're separated
Yeah.
by the mat.
Yeah, but then
net.
we shouldn't put more there. I think that's it for that bio box, right?
Otherwise,
It's because it's going to be quite muddy.
this will be
Yeah,
a
I
lot
can give
of
it a
mud.
shake, but not out. Like I'll let the sieve of the holes do the sieving.
Yeah.
So if you just
Yeah.
come left with me a little bit, we'll just get out of it so it doesn't totally obscure or vision.
You're doing a great job,
Yeah.
Jenny. This saves us hours.
And clean windows as well.
Well,
So we're trying to get rid of the really fine sediment. This is something we usually do on deck. You usually do on deck with the sieves.
it's necessary to get the small specimens in there, but we have a lot of samples where the small specimens are actually kept in the net, and these are attached to a coffee mug, so-called, where you can see the handle, the bottom left. Okay. Ja, klar. Alles klar. Wie du möchtest. Und dann tauschen wir dann immer, wo du kommen möchtest. Oder wenn einer von uns eine Boot getroffen wird, das ist ja das andere. Ja. Okay.
So we're good at that pack though, is that?
Yeah, that's good enough. And that is, yeah, the bottom left. As you can see in this drawer, we have 2 mugs. They've already been filled. We have a sponge on the right, and we're putting now our scoop contents on the left, bottom left compartment. Perfect. Splendid. Thank you.
Thanks a lot.
I think that's it. A little looky inside. Okay. Andrea?
For me, it looks empty. Do you want to
Yeah.
check?
Well,
if
keep
this
them around.
one's-
Looking good.
It looking good, yeah, completely empty.
Yeah. Okay. Let's throw that away, so.
Yeah, can store everything. Thanks a lot.
Okay.
Yeah.
Standing.
So after this is stored, we can fly on.
Yes.
Leaving confused amphipod. We often look to our phones because we get certain sampling requests
Yes,
from colleagues all over the world.
or comments on what we're looking at, and
But sorry, we can't sample that much.
but I mean the layer of pebbles is really thin. It's just one layer of pebbles, then it's mud.
Mm.
And I don't think we've ever seen this type of ground yesterday.
No, we haven't.
This is a bit new.
Zero, more or less.
Okay.
I'm missing Bodle sauce and
Can we start?
yes, please.
Yes.
Start please.
Starting going, is it? Yeah. Bridge RV, can we resume the move there, please?
Doing move 000 at 0.1.
Yeah.
Our pilot is now Andrea and co-pilot Patty.
Fish.
Temperature in the water is minus 0.4. It's a little bit colder than yesterday. I don't know what that is. If it's a lot of sponges or worms.
We're looking at this rock with encrusting units towards us. I don't know.
It looks like worms. It seems that every time you have a little surface, the worms really stick to it. But always on the same side.
On the same side, yeah.
Yeah.
Exactly. On the hand side.
Like here.
Yeah.
There's a hairy sponge coming up. That's probably a geodia. They are very abundant on the cliffs around. Yeah, there's a couple of geodias here. And the little soft coral. And then the orange colors you see are little anemones. The pink ones as well.
And a whelk, like a snail sitting on there.
Oh yes, absolutely.
So, Pepper, the next one, shall we just stop in front of it and have a look?
Mm-hmm.
Might be a bit late now.
Yeah, Bridge RV, we started point one.
Noise 0.1.
Sometimes we see this dark brown patches in sediment. From looking at samples this morning, there was a lot of rhizomes, but this doesn't quite look like this. Do you mind stopping here so that we can have a closer look? Thanks. Thank you.
They do seem like, because this is the bryozoan, I think, and then here are dead bryozoans.
So the dead bryozoans will be the brown ones, and then you have the white little ones living on top. So bryozoans are lace animals.
Yeah, they are. Yes. Also filtering the water and eating in the water column. It's a large group. They're usually very small animals, but they're very important. They're everywhere.
And there are very few specialists. Well, there's a very nice sponge, the white one. And
It's
this little amphibot just ran behind it. I was hoping it would come to the front. It just disappeared. So, an enemy.
The little round sponges look like golf ball sponges, but they're not. They're young geodias growing, which means that they like actually to settle on this mat of, maybe it's a mix of spicules and bryozoans and so on.
Okay. Shrimp. We can continue. Thank you. Yes. Have you looked to see stuff?
Nope.
RB bridge, let's resume the move, please.
So constantly we have to talk to the bridge on the boat so that the boat follows the
No problem,
ROV.
thank you.
We are connected through a tight cable, a very strong cable, and the ROV and the boat move hand in hand. When the ROV stops, the boat needs to stop, and so on. And we are in the command center. We call it the ROV shack, where the pilots have a number of screens and cameras showing them a little bit all around the ROV. And we, the scientists, can see the main camera feed. And we can control that camera to the right and left, and we can zoom in or out to make our observations.
But when we zoom in while flying, you get dizzy. Have to avoid that.
But we're very lucky with the sea conditions, I have to say.
Yeah.
Outside it's pretty flat.
Okay.
Almost no swell. Meaning
No. No, let's fly on.
if the sea conditions are too bad, we cannot dive with the ROV. So for us, it is essential to have good weather conditions.
Can we stop in front of this rock, please?
Okay, bridge RV, all stop.
All stop.
It's quite incredible when you think of it that we are making a 65-meter long ship stop because we want to look and zoom in on little worms at 1,400 meters depth. That's quite a lot of logistics and equipment, which makes those observations quite
Lasers off, please.
precious.
Now, now what do we see?
Yes, I see
Some
amphipods, of course. I
Very
see.
good. I see sponges.
Yes. I think I see two amphipods.
Oh, there's one moving right
Oh,
across
yes.
the screen, just
Yes.
so you don't, it's not forgotten.
No.
And you see one sitting at the bottom of the rock
Yep.
on the left. And you see one like 10 centimeters on the right of the rock. You can, when they're alive from this group, they're often reddish and a shiny eye, so they can be spotted easy.
Mm.
These are relatively large ones. There'll be plenty more smaller ones.
There's a question I wanted to ask you actually. So I remember in the previous live you said there were 10,000 species of amphipods, but how many species in this area?
What is happening? What is happening with the light? We're not doing anything either.
Yeah, it's flickering a little bit.
Shall we fly on?
Let's wait.
Yeah, no, but
for them.
I think it's worse when we zoomed in.
Yes. Should we move on?
Yeah. You can turn the lasers back on, please.
I try to log really every fish we see because fish are one of the few groups where that can be identified on video. So by analyzing this video, fish experts will be able to tell something about the populations of fish on this unnamed seamount. We cannot do this very thoroughly yet for sponges because many of them that we see on the ground cannot be named. That's why we're trying to subsample some of them at least to try to get names so that people can analyze those recordings much better in the future. This fish is not afraid.
Ray Jarvie, can we resume the move, please?
000 and 0.9.
Thank you.
There's a little carnivorous sponge at the bottom, this little bush-like structure. But we've seen places where they were truly abundant on the cliffs of this unnamed seamount. That's a fish, okay. Wow. So we're meeting another cliff here. On this cliff we have two species of Geodia, Geodia parva and Geodia hancheli. These are typical Arctic species.
Now let's focus on the sea spider. Just here. Can we stop here? Yeah? It's just because it's right in the middle of the screen now. And they're so colorful.
Ray Jarvie, I'll stop.
Give it a look.
I'll stop.
So people have been asking on social media how large these sea spiders are.
You see the laser now, 10 centimeters. So what's it, 25? So the distance between the two pink dots is 10 centimeters.
Yeah.
And you can see the sea spider. The legs are it's
More than
larger.
10 centimeters, the legs.
Exactly. So it's around
Maybe 15? And
yeah, 15
yeah.
centimeters a leg. Your guess is as good as ours, yeah? And so across it's 30.
That's what I would say.
Yeah, it's quite large.
Which is very large
It's
indeed.
it's our lower arm.
Yeah.
It's from. And they're very pretty creatures, I think.
So, could you tell us, are these crustaceans or no?
No, they're not, but they're related to spiders. But when we have meetings talking about news, scientific news, then the sea spider people come to the crustacean meetings because they're more crustacean people studying the marine environment. So to learn more about interactions and influences from habitats and distribution, there's more similar methods to the people studying crustaceans.
So are these actually spiders that went to live in the ocean? Are they like arachnids? Are they part of the...
Could it be the other way around? Could it be that, well, I think that all life evolved in the ocean, but I have no idea if these were
if they came
First.
back or were originally
know.
in the ocean. Okay, wow. And is that a typical Arctic species, the one we're looking
Yes.
at? Yes.
Yes,
Mm.
it is. They also grow to the size in the Antarctic, but there are quite a few around which are much smaller and not as colorful,
Mm.
but not less interesting. So we'll do is just go down a little bit and zoom in here.
We can see actually that the worms are growing really on the pilolavas, basically. And that's where the geodias and the anemones are also.
And the Ascidians. There are quite
And
a
the
few.
Ascidians,
There are
yeah.
quite a few Ascidians here. Ascidians are this translucent objects with the two openings. Look a bit like a balloon with a hole.
Yes.
Still inflated and lengthy.
And all these organisms feed in the water column. They're attached to the bottom floor and they feed on whatever the current brings to them. For the sponges, it's really small things, really small living organisms.
Look, this anemone has just ate an amphipod.
Really?
How dare it? Here on our on the bottom right.
Yeah?
Yeah.
How do you know? Did you just see
no,
the amphipod?
No, I'm guessing because I just saw something light in there, so I thought it's the eye, but
Mhm.
I might be mistaken. And I have seen them doing. I mean, that's what they also feed on.
Hmm.
This is of the lower ones, just had a meal, and they're putting in their tentacles. That's how they feed. Their mouth is right in the middle. We're not actually moving anything with our camera, it's the rough moving, which is good.
But you can tell there's a lot of current. I mean,
Yes.
this is not current created by us.
No.
This is natural current on this wall.
I just wanted to look at the sequence again.
It's not moving that much, right?
No.
Is it
No,
because it's waiting for a prey, or?
Come on. You're asking
No,
me
but
the
I would expect,
psychology of
I don't know,
sequence.
that they're crawling on the floor
Well, there's something
with a
to do,
mission.
but give them a break. You know, just waiting for something to come along.
Mm.
Yes, thank you. We can fly on.
But look, there's the pilolavas, then there's
You can still fly on.
the tube worms, and then there's a crust of
Yes.
mud.
Yes.
It's like 3 layers.
Yes, but the anemone made it to
Yes,
hold on to the
to the bedrock,
yeah or yeah maybe
or
there's a little
even
bit
the
of-
mud actually.
Yeah, but it looks like holding on to the mud, but
Yeah.
it can't be. That's very nice.
It's very nice.
Some
Oh,
fish.
there's a fish here hiding. A lot of carnivorous sponges as well here. All the bushy white animals you see are carnivorous sponges, so they are not filter feeding in the water column. Instead, they are probably catching some amphipods or other crustaceans.
They will be. That's why these amphipods are so important, because they play such a central role of eating and being eaten.
Right, Jordan, I'll stop.
All stop.
So I'm gonna repeat a little bit for incoming viewers. We are on an unnamed seamount that is parallel to the mid-Atlantic ridge, but we're on the eastern side of the ridge, and this is a huge mountain. We're talking 30 kilometers long mountain, unexplored to this day, and we're the first ones to see the animals living here. There's a Echinoderm, let me type that. The summit of that mountain is not very shallow. It's at about 1,200 meters. So we're actually maybe 200 meters from the top.
Yes. We're 1,437 now.
Yeah. And one of the questions we're trying to answer is, are the communities on the eastern side of the mid-Atlantic ridge comparable to the communities on the western side that we've been exploring so far? And I mean, it's difficult to say right now, but there are a lot of common species. The geodias are one of those, the white glass sponges. But we've seen a couple of new players here, like the small soft coral, the worm-like mollusks called Solenogasters. So there might be some differences. Analysis will need to be done. Samples will need to be processed. Specimens will need to be identified. And we will tell you, but it will probably take weeks and months of analysis to be able to give a straight answer. And most of all, we will need to do genetics, because we want to see, we want to know if the populations we're catching here, subsampling, are connected genetically to the populations on the other side of the mid-Atlantic ridge.
I like all those fishes sitting in between.
Yeah.
And there's a shell, well, like a garden snail, related.
The lasers are 10 centimeters apart. So this fish is about 15 centimeters long. And you can see the geodias, those furry balls, are for most of them 10 centimeters, but some of them are definitely 20, 30, 40 centimeters large. And there was one paper 2 or 3 years ago in Nature Communication that tried to date the geodias, saying that a 20 centimeter specimen was about 200 to 150 years old. So these animals probably grow very slowly in the Arctic, and most of them are probably very old. So we can't just speculate on the age of the specimen that's 40 centimeters or 50. So these are maybe 500 years old.
Right, Jordan, can we resume the move, please?
This old age is also true for some of the hard corals we encountered.
Mm-hmm. Yes, that was during our bamboo coral dive a few days back.
And there are different methods of age dating, aging specimens. Some of the corals, they have like rings like a tree.
Oh.
You can really count.
Mm-hmm.
Yeah, count to the hundreds. So you know they're hundreds of years old. That of course only works when there's like a shift in seasons.
Just like in trees. Yeah.
Similar.
Seasons.
But
yeah.
for many of these invertebrates that we see here, we have no idea. Many of these actinaria, these anemones, we don't know how old they are or can be.
And amphipods, do we know how long they can? Are they annual organisms or?
No, but we don't know for the deep sea ones. There are also quite a few from lakes. It's like glowing, look. And those can be held for years in the aquarium.
Okay.
And in the deep sea, especially in the Antarctic, they can grow very large and they have to molt. So they get out of their cuticulars, like skin, that's limiting their growth. And then they're very vulnerable directly after hatching. Hatching the wrong term, but
molting.
Get out after the molt.
So we don't know, but probably several years.
Yes, many years. Definitely.
Have people been able to raise them in aquarium?
Yes.
Wow, like reproducing.
Amphipods, yes.
Yes.
Yes, for sure.
Deep sea ones, or only shallow?
There are attempts from deep sea ones, but when they get too stressed when sampling, they sometimes release their juveniles. So they haven't come into the next cycle of breeding. But the shallow water ones, that has been done quite successful.
Okay.
They're also used as fish food. So there's a commercial interest in these.
So I have to say, for deep sea sponges, it's very, very complicated. I don't know of any aquarium like open to the public that has really deep sea sponges to show, or they're not very, very deep sponges.
Most of the amphipods are too small to be shown in aquaria to raise public interest.
You need to get the really
yes,
big ones from the abyss. Yeah.
They don't have a swim bladder, so they don't mind too much getting up. Well, and that's also the wrong term, but they survive. Our difficulty here is to keep them cold. So we keep them in cold seawater. And we have a photographer on board who's able to take fantastic pictures. And the specimens still have their live colors.
What's that?
And have all the appendages intact.
Yes.
His name is Solvin.
Yes.
He's doing a great job. So we're trying to avoid like getting air bubbles into a sponge, for example, on board.
Yes.
Keeping the animals cold is essential.
Yes. So when the ROV comes back on deck, we usually prepare buckets of very cold water to welcome these animals and try to keep them alive a little bit longer, so that pictures can be taken, observations can be made while they're still alive. We also want to preserve the DNA as long as possible, especially since we're collecting also for RNA. RNA is a molecule we all have, but it's a bit more fragile than DNA, and you have to sample it ideally when the animal is still fresh. Exactly. So when I put a sponge in alcohol, it will extract a wide array of compounds. So I keep that ethanol, I call it the first juice, the first extract. I keep that first extract preciously because it can be studied for all the molecules it has extracted. We can look at the different masses of these compounds and maybe find something new.
Speaking of ethanol, this is a dry ship. And the ethanol that we've got, well, it's poisoned. It's wrong.
Oh yes.
So chemicals, compounds are put to it that it's good for preserving, but you wouldn't be able to have any
to
yourself.
drink it. Yes.
Are the lasers on?
Yes, yes.
They are. They just
So
downhill. It's hard to see on this one.
So we're in the blue, as we say, when we fly over a little bit higher because the terrain is either going down or is it a little bit complicated to handle, then we go in the blue and fly over to another place that's more suitable.
So overall we're following a transect, because while this area has not been studied before for any biological content, it has been mapped and to a resolution of 35 meters approximately. Yes. So we have some unforeseen dips here.
And we were talking about chemicals extracted from sponges. So there's actually several drugs on the market that are either inspired or that have been extracted and discovered from sponges. Two drugs are currently on the market called vidarabine and cytarabine, used as anti-cancer, against leukemia, lymphoma, or also against herpes viruses. And these drugs were extracted from a sponge from Florida, from the tropics, in shallow waters. So this is just to give you an idea of the importance of doing chemistry analysis on sponges. They can lead to really interesting drugs for us.
Yeah, and not only drugs, they're also used like in the anti-fouling industry.
Yes.
So there's a lot of research going on.
Yes.
What can be extracted as a natural compound.
Yes, exactly.
Like to put on boats so that other things don't settle on the boat.
Yes, it's an opportunity to find natural compounds from nature that we could integrate on boats to prevent fouling. What we call fouling is when other organisms grow on the hull of the boat. It's a huge problem in boat industry. A lot of money is lost because you have to clean regularly the boats, the hull of the boats, because when things grow on the hull of the boats, it slows them down and you lose money, of course. If your boat
You require more petrol too.
you need more gasoline and so on. So there's actually a lot and compounds extracted from acidians or sponges that are currently tested in boat paints.
And sea cucumbers.
And sea cucumbers. I mean, all these animals also don't want anything growing on them, so they have already developed through evolution.
Some of these sponges don't
compounds.
mind, but some are very clear.
Yes, yes. So yes, there's a lot of biotechnological applications for chemistry extracted from marine organisms in general. We call it the blue economy. The blue economy is everything that basically people can make money out of the ocean, and this includes all this blue biotechnology field.
Well, sponges are used used commercially as bath sponges. You
Yes, that's probably their oldest application.
Yes, the ones without any spicules.
Yes, since the Greek times, sponges have been used, and that's why there's a word in Greek for sponges, in Latin as well, spongia.
Jorvay, will you increase point 2, please?
The Latin word for sponges is porifera. That refers to all the pores they consist of.
Yes, and the pores are there because they need to filter water, and so they need for the water to come inside and also to push it outside once they've eaten what was in the water.
It's a bit strange how they eat. I mean, they don't have any organs.
Yeah,
They just have pores.
they just have pores. There's no
Different-
organs.
different kind
That's
of
true.
cells.
So no digestive system at all. Just canals bringing the water to special little chambers, and in those chambers they have special cells that will pick up the food, not just any food randomly, but they really select what they want to eat, and then they will just let go everything that they do not want, and yeah, it
Often
works
through one opening.
very well. Sorry?
They let go through one opening.
Yes, then the water gushes out from what we call the oscula. That's the main opening through which the water comes out of the sponge. There can be several of these main openings, but usually when you see one central hole in the sponge, that's the oscula.
They've been around forever.
And they've been around, so not as much as
Not as
bacteria.
forever, sorry.
Not as
But-
much as bacteria, but
dinosaur level.
yeah, way before the dinosaurs. So the dinosaurs appear in the Trias, the first dinosaurs, and sponges appear about I would say 300, 400 million years before that. So they are they emerge way before the dinosaurs. But they were also living at the dinosaurs' time, the Geodias. So for instance, so one of my studies, I looked at Cretaceous fossils of Geodia. So they were there during the Cretaceous and the Jurassic, probably. So Geodias were already present in the seas, side by side with dinosaurs.
So they're fossils that were found, or how do you know?
So we have two types of fossils for sponges. We have fossils from the spicules, so which is the skeleton, the glass skeleton of sponges. And we also sometimes, more rarely, we have actually whole sponges that were fossilized. But it's a peculiar sponges. It's what we call the rock sponges. They're very hard, and therefore they fossilize much better. Oh, there's a nice fish. So the fossil record for the rock sponges is very good. And these rock sponges actually start to appear also during the Jurassic. We have actually sponge reefs of rock sponges at that time. They're very important in the Jurassic underwater world. Not necessarily. I think the white spots are probably the one we collected earlier. Maybe some are snails as well, because I remember that we also looked at some in closer view earlier. There's a couple of carnivore sponges, like one right now, a little bit to the left of the screen. But it's essentially paleolavas with a bit of mud.
Covered in mud.
Yeah. And are we going down? Yeah. So we have a bit of a passage going down now.
Finished.
Okay. Let's try to plan a transect going up, because we know it's much better for viewing and stopping and exploring in general for the ROV. But for this unnamed seamount, we have maps with a resolution of 35 meters, meaning that every 35 meters we have pixels basically that are the size of 35 meters. And although that may seem quite precise, for other maps we have 1 meter resolution. So of course we know far less, and it's much more difficult to plan a transect here in this unnamed seamount because we have this uncertainty.
I would actually like to stop and look at this.
Next to the sponges?
Yes.
You might see us regularly looking at Andrea's screen, because she has a screen that shows us the below the ROV, which is really useful because it's good for us to know where she can land with the robots.
Or just hovering. You don't have to land. It's more stable landing, yeah. See a little shrimp here on the right, in pink. Don't worry.
A lot of crustaceans in the water. It's very rich.
And also several shrimp sitting here. See the pink ones?
Mm-hmm.
And little amphipods. And all of these. Area. Sorry, I'm zoomed out.
Are these amphipods sitting on that sponge? Yeah.
I would guess so, but I'm. Yeah. I can't see close enough to confirm, but yes. I counted 11.
There's a little gastropod, but basically I'm seeing the same little sponge species that we've previously collected.
I'm trying to center it, but I can't move up anymore.
Oh yes, okay, I see it.
Some anemones feeding posture. Others have their tentacles in. Yeah, we can fly on.
Sure.
Thank you.
Wow, look at the amount of
Stuff.
Yeah.
Sediment.
But a lot of living crustaceans. This is a very rich environment, which should attract a lot of fish and squid and predators. So the water temperature hasn't changed. We're still at minus 0.4 degrees at 1,450 meters depth. I was discussing with the classroom this morning, and I had another interesting question. I had loads of interesting questions, but one of those was, how do the animals get their oxygen? And it is not obvious that when you're very far away in the deep sea that there's oxygen, but there is. It's in the water.
It's essential.
Yeah. Without oxygen, none of these animals could live. But then you use different techniques. I mean, the crustaceans will have gills, like the fish, and for the sponges, they will get it through diffusion. So the water coming in their canals will be water with oxygen, and that oxygen will get into the sponge, and it will be brought to the cells, directly to the cells. But yes, in all of this mass of water, there is a lot of oxygen.
And in this part of the world, it's also a lot of salt.
Yes. Yes.
Bridge RV.
That's why it's so called heavy water.
Please.
It's quite salty. And the different water layers, the more salt, the colder, the denser it is, and the heavier therefore it goes.
Exactly. It's not known, maybe very well known to the public, that water can have different weight depending on its physical properties. So depending on the temperature, like you said, and the amount of salt. And cold water is more heavy, and salted water is more heavy, it's more dense. So it's going to go below another mass of water that is going to be warmer and with less salt. And
it's a conveyor system.
Exactly.
It's a
So
conveyor belt all around.
this creates
the earth.
currents. This creates currents, and
It's also our Gulf Stream. People will know that the Gulf Stream.
the Gulf Stream is well known to the public, and it is warm, less salted water.
So it can only be moved up because the salty heavy water moves down. So like a conveyor belt.
Exactly.
With
if you're-
1,456 meters.
Yeah. So if you're not familiar with the deep sea, it's not a homogeneous sea. There's actually different layers of water, and it's sometimes difficult for animals to move from one layer of water to another. There's different compartments, so it's not you're not as free as you think you could be when you're small, especially if you're a small animal and you're dependent on currents and water masses that are sitting on top of each other depending on their physical properties.
Sometimes the horizontal distribution is much wider than the vertical one, so they can be, well, speaking of amphipods here, they can be found 1,000 kilometers apart, and even the same species in the Pacific and in the Atlantic, which is quite impressive.
It is.
That they're brooders, so they don't have larvae.
Mm.
But they are not found in like 500 and 4,000 meter depths. So that they have a vertical limit, for example, between 2 and 4,000.
Yeah. So I'm going to say it's exactly the same thing with mice sponges. We have the same phenomenon with widely distributed species if you stay on the same water mass or depth, but as soon as you would go shallower, you'll have different species. So these boundaries are real.
We only look into such selected samples that there are always these questions, how do they get there? But maybe they're everywhere in between, but there are way too few samples to say that.
Mm. Yeah, we've explored only a very, very small part of the deep sea. So I've seen numbers like we've explored 10% of the deep sea, which is nothing. It's maybe even less. And if you consider the deep sea as more than half of our planet, well, more than half of our planet has not been explored at all.
I think a little bit of different numbers. Think of the Earth as 3/4 in water, and 90% of that is considered deep sea. So everything below 200 meters is deep sea.
Yeah.
And then it's less than 1% explored of that vast area.
Mm-hmm. Mm-hmm.
So explored to a level that you know what kind of species live there. We're not talking mapped, we're talking biology.
Mm-hmm.
That's why it's so likely to find species that haven't yet been described, that are new to science. When we look at the deep sea small stuff, especially.
Mm-hmm. It's difficult to access, and there's actually few scientists, few specialists for every group.
A so-called taxonomist. Well, it takes time, and it's not taught at universities a lot.
Mm. Yes, I would say a lot of genetics and genomics have been kind of replacing the older knowledge, or the zoology and the botany courses that used to be more common before.
But what we're doing here is also creating baseline data. So for further studies, we have images, specimens, and the genetic reference. So when it's found somewhere else, then you can compare the DNA to know if you've got the same specimens.
And it's very important that the specimen, before being sequenced, has been identified by an expert, so that the genetic sequence going on internet has been really verified, so that you can trust it, so that it becomes a reference sequence.
But these open access databases have improved massively over the last years. Last 10 years. The museums, with their reference databases to what's there, as well as the genetic databases, a lot of them are open access.
Yep. Yep. And that opens the door to environmental DNA, because environmental DNA is extracting the DNA from the environment. It can be sediment, it can be water, it can be air, it can be an animal, and picking up all those bits and pieces of DNA in there and sequencing them and trying to tie them to a species name, trying to see from which organisms they come. But to be able to do that, you do need a reference database.
So there's no point having a lot of sequences, and you know, oh, these are 20 different things, but you don't know what they are.
Absolutely. Yes. So our job as taxonomists is making sure that the reference databases of our organisms are correct. And for that, we need to identify and work hard to make sure new species are described as well.
I once was invited to a workshop, and this is the barcode of light live databasing,
Yep.
so just to get good reference points, so to look at them taxonomically, and these were for amphipods from Norwegian waters.
Oh.
And while doing that, we found a new species,
Okay.
and we described it, and we named it for Unnamed tandberg, who's
Mhm.
also on board, expert, and it's quite nice that this species is now found quite often.
Okay.
So once it has a name and a reference, and
Then people can identify it and find it. Absolutely. Otherwise, it's invisible.
yes.
Yes. Naming animals and plants, any living organisms, is essential to make it visible to science, to the public,
Yeah, and for-
to policymakers.
it's very barren here.
Yeah, and we're still going deeper.
Didn't you tell us it's soon over this downhill path? Almost there, they say. It's a bit like
So we're almost.
kids on a long car drive. Almost there. Go. I see a lot of blue.
So again, oh, two fish next to each other. That's
Sweet.
a family.
No.
That's us guys at the end of the line there.
Okay. Now what do we do?
Is it three fish?
Yes.
Oh
Let's get
my!
them. Look there. Look. Ah,
Oh, are there others?
no, I think these are sponges.
Oh, okay. But still, three fish next to each other, that's something. It's true.
Can we go up sideways? I'm not seeing this wrong. We're at the end of the transector.
We're at the end, yeah. We've come down.
So what is the best view
Best view for
for looking at this camera? Looking at the to go
turn around.
turn around and up. But then we fly over the same area that
Yeah.
we've just
The Geographic, I'll stop there. I'll stop.
Can we sit down
Sure.
here? Yeah. Just turning around.
Do you
Yeah.
mind if I go into the dry lab and try to get some information?
Shall I go? Do you keep on talking?
I can keep on talking, no worries. Yes.
And do you
So,
have a preference for sampling?
no, not necessarily. Okay, so we've arrived at the end of the transect, and we need to make a decision on where to go next. We're going to consult the cruise leader and other scientists that are looking at the maps as we speak to try to see which part is now a priority for us. And these moments are perfectly normal in exploration because we sometimes rely on what we're looking at in real time to decide where to go next. Could you enlarge a little bit the map?
Larger or squeeze it out?
Squeeze it, yeah. Yeah, thank you. Is it continue? Does it go down, 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 or probably?
doesn't give you anything beyond
There's
100 meters.
at 50 away. When we're coming along the top here, when we're back here, there's nothing. And as you can see in the shading, it's getting down. So it's broken anyway.
But this part might be a bit higher.
Maybe there, yeah. But you gotta go down here. Come up, go down.
Yeah. And how far are we from this part? Okay. Nice. Could this be like a blue, or maybe we have information? Could be. We finished the dive here?
Yeah.
Okay.
Shall we do some more sampling? We've still got...
Bring back a tea when you're coming again. No rush.
But so if we want to do sampling, we would need to
Sorry, no.
turn back, like Patty suggests, because we would need to be uphill, I guess. But I don't know if what we've seen so far.
That has been a bit uninspiring.
Yes, because most of the little sponges we sampled.
We already sampled.
Amphipods we did.
Yes, I would have loved to get some of the Solenogasters that they had, but I didn't see any.
What about push cores? Should we maybe try to get push cores?
No, they've already taken those.
Okay.
Just before.
Should we
just turn and see? Just
exactly.
turn and fly a little bit to see if the view is completely different when going uphill, and then we'll finish the dive.
Yes.
Is that okay, Paco? Paddy? So,
To go
is
back
it-
over the line we came.
Yes,
Yeah, or-
a little bit.
Yeah, or-
and see if we can sample something else.
The other transect is too far away. It's 2.5 kilometers away, so we would finish this dive, but we are ahead of our time
Yeah.
because there wasn't so much to see. So
Yeah. That's a 180 on the heading. We're putting a big twist into the.
do you think the visibility would be better flying uphill? Yeah.
It will be, but the problem is to do 180 degrees. You're putting a twist into your underlip.
Oh,
Yeah.
but we can do very slow.
We're facing the slope here. If you look up, we turn to our right. If you look up at your camera back up,
Yeah,
tilt up.
yeah. So that's where we come from.
Well, no, well, pretty much, yeah.
Yeah.
And we turn to our right a little bit.
Yeah. I mean, it doesn't look very promising to me. We might as well end the dive. I mean, we do have
We end the
slurp.
We end the dive. Let's have a dive. We're just so surprised. Normally it's always the other way around.
Yeah.
You know, it just seems a complete waste of not having some. Do you want to sample?
I mean, we could take scoop
Yeah,
a bit of that, and that would be
yes.
sorted. We do
Yes.
that. Would it be possible to scoop something in this material here and put that in a box on the starboard side?
We could try, depending on how high up, just tilt down on your camera.
Yeah, sure.
It's quite high.
It's quite high and quite steep.
Oh yeah, it is. It is. Okay. It's always a bit.
Yeah, it gets very steep there in that patch.
It does. Oh, and look at that one. That's maybe a bit closer. This. But it is. Okay, I see. Oh yes, I see. It's steep.
Yeah, it's doable.
But could we settle? Could we settle here and collect stuff here?
Yeah, you could, yeah.
Okay, let's try to do that. So of course we plan our dives with sampling in the middle. Here there was not so much to sample, and therefore we finished the transect earlier than
But
expected.
It's very unusual for us because normally we stop all the time, but there's been so few well, communities that haven't been explored by the team also before us.
But it's part of exploration. It's a lot of unknowns, and you have to adapt.
Yes. Yeah, no.
But so we're going to settle here. I saw there was a little
Wherever
place.
the roof can settle.
and
this,
we
then we can
take
live with.
2 things and then we're off. Yeah, we've been keeping going down like the last 100 meters.
I know.
Okay. It's always a bit tricky. We have to leave space for the boxes to open, the arm to move. It's very tricky, especially on a steep slope. So we'll see what can be done. Let's zoom out as much as possible. Are you interested in this? I don't think we've collected that species yet.
No,
Maybe for solvent? It's a
we
nice
can
color,
try.
right?
We can try. Is it slow?
Oh, it's gonna be, no, because it's gonna be, does it produce mucus? Because I mean,
so.
it could be all along there, it's gonna be really good. And
Yeah. But
it's one of those, I think we've already had one.
not in purple.
Not in purple, so, yeah.
That's the only thing to see.
Okay, we're thinking of sampling the sea star, the purple sea star. Yes. On
Can we
the-
take it with a shovel?
Yeah.
And then we get some of this mat with it.
Yeah, and then I think this is Lysodendrorix, so maybe first I can zoom on it.
Let's zoom a little bit.
Yeah.
And just a sec. Can we stop? Can we now have the lasers off, please?
Off.
Yeah, perfect. Because then I can collect this for Sergi,
And I want to collect this.
and you want to collect this.
Of course,
No,
that's why I'm not zooming any further.
No, but then we can put
That
a
makes
few
it-
more things in the box.
I think we'll, yeah.
Yeah. Let's make a little close up before we. There.
Mm. Yes, can we take the arm?
The purple one, is it?
Well,
Yes, the purple one. Yes.
the purple one and a bit of this one. So it can it all go in one box?
Yeah.
Like in one scoop, or we try several?
Oh no, you have to do 2 different scoops.
Yeah,
Two different
that's
scoops.
fine. That's
Okay.
in the same.
So that would be cast.
Probably
Probably cast 15.
I'll bring it in.
Should be feeling them.
For sure.
Went faster than I thought.
Yeah.
Well, we only went downhill.
You'll get that now, won't you?
We will sample that one now. We will sample this one, the sponge, and maybe that together. So because we
Could you bring your camera around?
Yes, absolutely.
To prepare for the next one.
and can we
Have we got confirmation?
nothing. There's no answer. There was. It doesn't matter. We're not. But I think it's
stop. Yeah. I will let you know.
Thank you. So we're using the scoop to try to collect a few animals here. We're at 1,475 meters depth.
Way off the boulder.
76 degrees north.
Yes. The sea star is still there.
Yep.
Sorry, my eye off the water, sorry.
It's probably actually sitting on the sponge, so it's probably a bit soft behind it. Just to let you know. Yeah, I think. Yeah, it's in the box. Yeah, I saw it going down.
Yeah.